for her welcome address.
Thank you, Dr. Monica. A very warm good morning to one and all. Without further delay, this is the fifth session of the professor's presentation, which had started yesterday. And today we have the professors of 4 disciplines: floriculture and landscape architecture, fruit science, post-harvest management, and vegetable science, who will be presenting the significant achievements of the postgraduate students' research work of the horticulture discipline. And as Dr. Monica has already mentioned, that we have with us Dr. Major Singh sir to chair this session. We welcome you, sir. So, so without further delay, now I request Director sir to kindly apprise the audience about the chairperson of today's session. Thank you, sir.
A very good morning to each one of you. Dr. Major Singh, member ASRB, Dr. Anupma, Dr. Brahmanand, distinguished colleagues, students, ladies and gentlemen. We are here today to have the presentation of the significant research achievements by the students of horticulture sciences disciplines, which will be presented by the respective professors. And I have great pleasure in welcoming Dr. Major Singh as chair of this important function. As you all know, he is currently member plant sciences in the Agricultural Scientist Recruitment Board. And Dr. Singh is a very distinguished vegetable scientist, both in biotechnology as well as in breeding. He served initially at the Indian Institute of Vegetable Research, a very long stint, and then was project coordinator, director of National Research Center on Onion and Garlic, and then he joined as member ASRB. He has been bestowed with the ICAR Team Award for Outstanding Contribution in Vegetable Breeding, Rajiv Gandhi National Award for Ministry of Home Affairs, and Dr. Harbhajan Singh Award for Outstanding Research. He has served as Vice President of Indian Society of Alimus, and also he has been involved in research management positions for very long, brings a very rich experience. He has written 3 books on heterosis in vegetable crops particularly, published more than 200 research papers. He is fellow of National Academy of Agricultural Sciences. But more importantly, if you look at the list of varieties that he has developed, it's amazing to see, and those names are so pleasing to listen and to read across the vegetable crops, like Kasi Sandesh, Kasi Komal, Kasi Taru, Kasi Prakash, and these are in different crops. These are brinjal varieties, 3 hybrids, Kasi Surk, Kasi Sinduri, Kasi Gaurav, and 1 variety, Kasi Anmol in chili, 2 varieties, Kasi Shakti and Kasi Mukti in pea, 1 hybrid, Kasi Abhiman, and 1 variety, Kasi Aman in tomato, and 1 variety, Kasi Pranam in French bean. So you can look at the list of varieties that he has developed, and these varieties, in fact, if you look at the practical significance of these varieties, if you go to Varanasi and the whole of eastern UP, there has been a vegetable revolution sort of. In that area, if you travel, the kind of prosperity it has brought to the vegetable farmers, and those farmers are not very big farmers, the farmers having less than an acre, half-acre land. But if you look at the peas cultivation, and during this season, starting from early September and then till December, the pea cultivation has brought a huge, a revolution in that particular area, and that is directly related to the prosperity of farmers. So he is a big achiever in terms of the research that has impacted the well-being of the society at the grassroots level. We are pleased to have you, sir, as chair of this session. He was also my senior in Banaras University while doing PhD in genetics and plant breeding. So I would like to extend a hearty welcome to you for being here. Thank you so much, and we look forward for your kind comments and suggestions so that we can improve our program further. Thank you.
Thank you, sir. As convener of the session and with the permission of the chair, I would wish to apprise the House regarding the modalities for today's presentation. For this session, that is session 5, we shall have 4 presentations from the professors of the following disciplines in the following order. First will be floriculture and landscape architecture, to be followed by fruit science, then post-harvest management, and then vegetable science. For all the 4 disciplines, the allocated time has already been conveyed, and it is requested that since today we'll be having award lectures from 2 p.m. onwards, so kindly restrict to the time given. For the ease of the presenter and also to help you wind up your presentation, a yellow light will blink before the 2 minutes time before the end of your presentation, so that you can wrap up your presentation, and the red light will blink when the allotted time is over. So kindly be precise, and as we have been doing yesterday, so that we are enabled to finish in due course of time. With this, I now hand over the session to the respected chairperson for conducting further proceedings of the day. Thank you.
Thank you very much. First of all, I must thank Dr. A.K. Singh Saab for inviting me to chair this very important session, because IRI is our premier institute, and to interact with you is really honor for me. So I will invite one by one. The first presentation will be on floriculture and landscaping. So I invite the person who is going to present. क्या नाम है इनका? Professor K.P. Singh, please.
Hello. Good morning to everyone. Most respected chairperson of the session, Dr. Major Singh Ji, member SRB, Honorable Director IRI, Dean and Joint Director Education IRI, Convener Dr. Joshi, Project Directors, Head of Divisions, Professors, Scientist Fellows, students, ladies and gentlemen. I am going to present the significant educational achievement in the division of floriculture and landscaping. Actually, our discipline as present is division of floriculture and landscaping, but the students who are going to get degree, they got admission in division of discipline of floriculture and landscape architecture. Actually, that is confusion. Both the terms are being used. Now, new student next year onward, they will be getting degree in floriculture and landscaping, as per the change of this discipline in SRB and everywhere. You see the position of faculty. This program is being run at IRI and IHR Bengaluru, and we have a total 22 faculty, including both the centers, and out of that 18 are research guides, and totally we have 82 scientists, including both the centers. Coming to the list of MSc degree recipients, there are 6 students who are going to get degree. All are from IRI New Delhi. This is including along with their chairman. This research was divided into 4 research theme areas, which is listed here. And coming to the significant research outcomes theme-wise, this work was carried out. First one, this in vitro response of French marigold by Chetra, under guidance of Dr. Sapna Pawar, and she took some genotypes of marigold and screened under in vitro conditions against salt tolerance using this NaCl 50 to 200 micromolar. And she got that selection FR5, R-5 got best one, performed well, followed by that variety Dentimerita, so that we can use it in our further breeding program. Another study was carried out by Kusma MB on chrysanthemum. She took 50 varieties on dot tolerant in chrysanthemum. She took 50 varieties. Experiment was conducted in pots and two stages. First on vegetative phase and second on flowering phase. Stage were imposed dot tolerance stages, and she got that one more variety performed better with regard to this dot tolerant in chrysanthemum. Another study was carried out, Mr. Balaji, on turf grass management. This is a new area we are taking up. And work was carried out to use the wastewater, which is treated one, to find out the best grass. And his study revealed that the three grasses, Barbuda grass, and this Joycia grass and seesaw grass, they are found better with regard to vegetative growth, absorption of heavy metals, and other parameters. So these can be recommended for this lawns. This study was carried out with Sangeeta Roy on rose to screen the rose genotypes against salt tolerance. She took 5 varieties, 3 from loose flower category and 2 from garden display. And her studies revealed that under garden display category, Pusa Lakshmi variety they performed well up to 100 millimolar. And under loose flower category, the variety Rosa Chinese strain FLS RC1 performed better up to 200 millimolar. In this theme, Mr. Valaras, who has this, he worked on Lilium at regional station Katran. His chairman is at Katran only, and on Lilium for this dormancy problem is there in Lilium. When we are not having the storage facility, then how we can use our this freshly harvested bulbs for without storage to break the dormancy. His studies were revealed that soaking of bulbs, freshly harvested bulbs, with GA4+7 and 5-cytidine for 24 hours, they exhibited early bulb sprouting and flowering, whereas the application of CPPU and metatoplin resulted in better bulb production in Lilium. Another aspect is micropropagation of flower crops. Under this, this Mariam Firdaus worked on this, and this is a new genotype, although it is an old one, but we have included in our research program. Recently, this is Tagatus minuta, which is very tolerant to low temperature. It's grown in Himachal Pradesh. To keep it in our breeding program, now first we are developing the protocol so that we can use in advanced work like protoporous fusion and their mutagenesis, all that under in vitro conditions. In view of that, this protocol was developed, and she got that among the 2X plant, this shoot tip gave better results as compared to this nodal X plant, and she also standardized media, and among the variety TM2. This genotype gave the better result with regard to all tissue culture parameters. Again, this is a list of degree recipient of PhD students and along with their chairpersons. There are 9 students, 2 from IHR Bangalore and 7 from IRI New Delhi. Coming to this significant achievement under improvement, this theme, this work was carried out on induction of double haploids. Actually, in view of this, to use this technique to develop breeding lines for different regions like this for summer season, cold, where we don't have any variety. This study we are taking up. And the first variety, this work was carried out by this Srijana Pradhan to develop the protocol using this Tagatus erecta and Tagatus putida 2 species. And she developed this regeneration, and this ploidy analysis was done through this chromosome counting and this chloroplast counting. And after this, which were doubled also, this haploid after screen isolation of haploids, they were doubled using this originally, and double haploid was also hardened and transmitted the field. So that through this study we could standardize the protocol for regeneration and isolation and doubling of the DH lines. Using this protocol, another study was carried out to develop or DH double haploids for heat thermotolerance, because we don't have any variety. For that we took 2 populations, one is Desi yellow and second is Desi orange. And with slight modifications, she developed again this protocol, and moreover, most important is she developed some DH lines which were transferred in the field and evaluated during summer months, May, June. And we got flowering so that they can be utilized in our breeding program, or if they are fit for release, they can be released also. And the next stage was also a conducted induction of haploids. Actually, in this study, main objective was to utilize the DH lines in breeding program. Using the previous protocols with slight modifications, these DH lines were developed, which were crossed with our existing male style lines, and these hybrids were developed. And this clonal fluidity was also tested using molecular markers, and which these hybrids are being assessed for in further generations. Another study was conducted on in vitro mutagenesis in rose, taking these two varieties, Pusa, Sarbat and Pusa Virangana, which are developed from IRE only under in vitro conditions, using gamma rays as mutagen. And we developed this protocol for under in vitro condition, as well as this standardized doses for this both the varieties under in vitro conditions, and we got some this mutant you can see from both the cultivars. Another study on production expert was conducted on marigold by this Tejasvi, and objective was during winter season, like you are observing now, we are having a lot of casualty in marigold crop, especially in December, January. How to save this crop during this period? We try to grow marigold under open condition using low polytunnel also for 1 month, nearly 15 December to end of the January, and along with some chemical treatments. This her study, you can see these are some field views, how this crop, the growth of the plant under low polytunnel and open conditions, they later kill a difference. And this her study revealed that the combination of varieties, this Pusa Bahar, is was better than the second one, with treatment of chito-oligosaccharide at the rate of 200 ppm and AM at sowing date of 15 October. Gave better result when used in combination with low tunnels to get the higher yield. Another study was carried out on landscaping. It's also new study. We are touching this also, and touching not only touching, we are emphasizing rather to have the more work on this landscaping, especially in relation to pollution control. So some species were taken by Mr. Lokender Singh, Azadiracta indica, Polyalthea, Ficus, Pongamia, and this Ficus infectoria, Acacia fistula, Alstonia, etc. And API and APTI were used to assess their pollution tolerance. And this study concluded that Azadiracta indica, Polyalthea longifolia, and Ficus religiosa were found intermediate tolerant to the air pollution, while these other species, Ficus infectoria, Pongamia, and Acacia alstonia, they were found sensitive to air pollution. This study was carried out by ICAR student at Sangeeta, and she developed a soilless system for production of gerbera using different systems you can see here. And her study concluded that vertical structure oriented toward east-west direction, deploying aggregate wick system on pot with AFC and this FIM and rice husk in 1:1 ratio, and modified nutrient solution formulation supplied at the TDS concentration of 600 ppm during vegetative and 1000 ppm during flowering stage was found superior with respect to plant growth, yield, and flower quality. Another study, this was carried out by Ms. Vaishali at our CPCT center under guidance of Dr. M.C. Singh, and major objective of this study was to how to regulate the flowering chrysanthemum. To meet out the demand of the market, whenever glut is there, you can stop your production, whenever demand you can increase. Having this concept, these light exposures were given, with the treatments are there. And you can see the picture, field view of this experiment. All treatments, some treatment are flowering, some are in bud initiation, some are in vegetative. At same time, you can have different type of this stage of crop. And this study concluded that stem elongation was there when more exposure was there to light, which is a desirable trait. And the fresh and dry weight accumulation was also there with the increase of light exposure. And supplemental light proved to be useful to regulate the flowering in chrysanthemum as per the market demand. Coming to the next, this theme is micropropagation. This study was also carried out at IHR. Earlier this student, she did MSc from IRE only, Ms. Sudamini, and she developed a protocol for in vitro lilium production and using bioreactors. And her study concluded that inner bulb scale is the best source of explant for in vitro mass multiplication of high quality plantlet using bioreactor culture in Lilium, cultivar Grand Paradiso, and farmers who could not afford cold storage facility, they can definitely use this bulb. As I told in earlier study, the dormancy is the problem, that they also try to solve this problem. This is a list of publications, research papers, training manual, and this chapters, abstracts, and these popular articles of the developed during the division last year. And this is a list of publications. We have the good publications in 7 and 8 and other NAS ratings journals. And these are some awards and honors received by our students. Dr. Namita got this gold medal, this Manmohan Atawar gold medal in floriculture from IAHS, and other students also received some awards. And these are the awards and honors received by our faculty member during the year. And this is a training program we organized for CPWD Mali, funded by CPWD. And these are some members, QIT members, and this ADG horticulture who visited our field, marigold field. You can see this marigold in white variety we are going to release soon. We visited and appreciate this one. And these are our students involved in very PG activities. The freshers welcome, this Teacher Day celebration, and our students also participated or rather served at Rashtrapati Bhavan as a volunteer during this garden festival. And I thank and for advice and guidance received from Director IRI, and Director HR Bengaluru, Dean and Joint Director Education, Dean, Joint Director Research, Joint Director Extension, Joint Director Administration, heads and professors, associate deans, faculty members, student SRFs. Thank you one and all for patient hearing.
Thank you, Doctor. Thank you very much for this excellent presentation and a good compilation of work done by you. Thank you very much. Now I will invite Dr. Manish Srivastava. He is going to present the achievements of Fruit Science Division.
Good morning to everyone. Most respected chairperson of this session, Director IRI, Dean Madam, Convener Dr. Joshi, other dignitaries present of the dais, Head of the Division, professors, faculty members, and my dear students. I am going to present the significant educational achievements pertaining to the discipline of fruit science for the year 2023-24. As far as the faculty and student strengths are concerned, we are total 12, out of which 6 are principal scientists, 4 are senior scientists and 2 scientists. And out of these 12, 9 are eligible to guide PhD students and 11 for MSc students. One of our senior colleague is going to retire next year. Currently we are having 48 students on roll, 37 for PhD and 11 for MSc. And this year total 16 students are going to have their degree in this convocation, 7 for MSc and 9 for PhD. Out of 7, 2 are from IRI Jharkhand, and in case of PhD, 2 are from IHR Bangalore. Whatever the work was carried out by our MSc student, that is grouped under different themes. The first theme is genetical studies and characterization. Ms. Pooja, Mr. Roshan, Mr. Shekhar Day from IRI Jharkhand, they carried out their research work under this theme. The second theme is reproductive biology, and Mr. Prabhanjan Bhanudas Rane, he carried out some work on grape. Third theme is crop production technology. Ms. Laya, Mr. Hasid, Mr. Madhumati from IRI Jharkhand, they attempted research work under this theme. So I'm coming to the first theme, that is genetical studies and characterization. Here Ms. Pooja worked with the topic studies and association between pulp color and seed related traits in guava. She took 5 parents and 42 hybrids from different cross combinations and characterized them for several pulp color and seed related traits. And she could conclude from her research work that 3 pink pulp and 4 white pulp guava hybrids, they are having better traits for pulp color and seed related, and 1 hybrid she identified having very high ascorbic acid content and 1 with very soft seed. However, she could not establish any correlation between pulp and seed parameter utilizing this population. Otherwise, in literature it is reported that there is a strong correlation between seed hardness and pulp color. One of our students, Mr. Roshan Kumar, who also presented for merit medal 2 days back, and he attempted work on characterization of a set of interspecific citrus hybrids orangelos. And here we all know that there is a problem in pomelo. Otherwise, it is a very good citrus crop with lots of nutritional value, but it is having thick rind and lots of seeds are there, and juice recovery is very less. So just to combining the traits of pomelo and this sinensis, in previous years our faculty, they have developed good hybrid population. So he tried to characterize that, and finally he could conclude that 5 Orangelo hybrids showed better quantitative and qualitative traits. Mr. Saket Dev from Jharkhand, he worked with the topic morphobiochemical and molecular characterization of segregating population of lychee under the able guidance of Dr. Mahesh Thakur, and he retrieved some SSRs and designed primer from the whole genome sequence data which is available in the public domain, and validated 50 SSRs, out of which 26 were polymorphic, and he could find 2 very informative SSRs having PIC value of more than 0.5. In addition to this, he also identified 6 open pollinated seedlings of lychee which are promising and may be tested and further released for the cultivation. Under second theme, that is reproductive biology, Mr. Prabhanjan Bhanudas Rane worked under the guidance of Dr. Madhubala Thackeray on the topic studies on pollen micromorphology, storage behavior and cross compatibility, and he observed, he did a very exhaustive work, and he could observe that there is a significant polymorphism for palynological characteristic among vitis species, especially if we talk about the presence or absence of colpus and the dimension of the pollen. And he could ascertain that pollen of grape can be stored for 30 days at minus 20 degrees centigrade, and for longer duration liquid nitrogen storage is the option. One more significant thing here is about the cross compatibility, interspecific crosses of Vitis parviflora and Vitis champagne. Vitis champagne is very important because it contains the blood for resistance for nematodes, salt and drought. So this type of compatibility, interspecific cross compatibility studies will be useful for future rootstock research. Third theme is crop production technology. Here Ms. Laya worked under the guidance of Dr. Jayaprakash on impact of plant growth regulators in relation to cold tolerance and flowering in papaya, and she took only 1 genotype under net house and open field conditions, and finally she concluded that application of thiourea 300 ppm and salicylic acid 75 ppm is useful in mitigating the negative effect of cold tolerance, and it also improved the physiological and biochemical parameters. Similarly, this salicylic acid 100 ppm improved the floral parameters. Ms. Madhumati, under the guidance of Dr. Vikada, worked on mulch, Tephrosia biomass mulching in mango, and she concluded that mulching at the rate of 7.5 and 5 kilogram per meter square was equally effective, and even they experienced increase in yield up to 25%. And the use of this Tephrosia as a mulch also improved the soil property, and it can be considered as a sustainable and cost-effective approach for improving soil fertility in less fertile regions. Mr. Harshit Kumar worked under my guidance just to see the effect of tree age and heading back in Amrapali, and after 2 years of experimentation he could conclude that young trees yielded bigger size fruits with better quality compared to old trees, and this effect is more pronounced after 20 years of Amrapali tree, especially under high density orcharding or medium density orcharding. So finally he concluded that trees attaining age of more than 20 years starts producing fruits of poor quality and must be subjected to pruning for rejuvenation and sustaining the production under medium density planting. As far as PhD degree recipients are there are total 9 students are there under different themes. In theme 1, evaluation and characterization, Megha, Nitin, Napika, Susmita worked, and for reproductive biology, Ashok Dhakar, and for mutagenesis study, Devyani M, Reena, and for abiotic stress management, Kripa Shankar and Mr. Sandeep work. First theme, evaluation of. So here Megha did very good work for understanding the dynamics of fruit fly in mango, and she concluded from her research work, very exhaustive work she did, and she could conclude a positive correlation between relative humidity and rainfall and minimum day temperature with the fruit fly population under mango orchards. And there are certain physical parameters, fruit physical parameters are there which are showing negative correlation with the fruit fly, that is peel thickness, sap volume and fruit firmness. And she also worked on volatile profiling and found that in case of susceptible, some specific volatiles are there like myrcene, pinene and ocimene, which is not up to that level in case of resistant ones. One of our students from ICAR, Nitin PS, under the guidance of Dr. M. Sankaran, worked on evaluation of pomelo, rosnees for bitterness and rind thickness, and he evaluated total 102 hybrids and 92 half-sips for different parameters, and finally he found 5 very promising hybrids and 2 half-sips, and he also identified through molecular research that 10 markers, very informative marker, and 1 marker he got which has linkage with the ascorbic acid trait. Ms. Napika worked under the guidance of Dr. Nagaraja on phenotypic evaluation and mapping of genomic regions governing yield and their component traits in mango, and she could identify total 28 QTLs in relation to 9 traits, and QTLs which were governing the vegetative growth related traits were located on different chromosome 3, 5, 7, 13, and those which were governing the flowering and fruiting traits were located on chromosome 4, 7 and 15 with significant log value. Ms. Susmita from ICAR worked on characterization of custard apple prosnies, and she evaluated a huge number of prosnies, more than 1000 prosnies she evaluated for different traits, and finally she found a very dwarf prosney, 38/17, which is having only less than 1 meter height, and she also identified good number of prosnies having erect growth and spreading growth, and 56 prosnies she identified having stable self-fruitfulness with good horticultural traits like more than 30 fruits and good fruit size and TSS. And she recommended 2 hybrid prosnies, 10-3 and 14-2, which are on par or superior to Arcasahan, the existing commercial variety. Second theme, reproductive biology. Mr. Ashok Dhakar worked under the guidance of Dr. V. Patel on the topic studies and floral development and reproductive biology in grapevine. And the interesting observation of his thesis research work means conclusion is that he got some research evidences that the berry and seed traits in response to various pollen sources clearly demonstrate the existence of genia and metagenia in grape. And he recommended that beauty seedless is an important parent if we are looking for berry related traits for improvement. He also worked out on sequencing and then identified some of the differentially expressed genes which are related with the flower development and regulation. Third theme is mutagenesis. One of our student, Ms. Devyani, under the guidance of Dr. O.P. Avasthi, worked on in vitro mutagenesis in Kinno, and she did excellent job as far as the standardization of in vitro regeneration protocol is there, and she worked on standardization of in vitro somatic embryo, then somatic mutagenesis, determination of the lethal doses and validation of the mutants. And she finally concluded that the direct somatic embryogenesis system is the most efficient system for gamma irradiation and EMS induced in vitro mutagenesis. Another student under the guidance of Professor O.P. Avasthi sir worked on in vitro mutagenesis on another crop, that is sweet orange, and she finally could standardize the protocol for in vitro mutagenesis in case of Mosambi. The last theme is abiotic stresses. Another student under the guidance of Professor O.P. Avasthi, Mr. Kripa Shankar, worked on salt tolerance behavior of sweet orange, cultivar Pusa Sarat, and he concluded from his research work, he concluded that Pusa Sarat grafted on Cleopatra mandin C47, X9, N1 and N3 root stock demonstrated greater level of NaCl tolerance. So these root stock may be recommended for sweet orange under those areas which are having salinity up to the level of 60 millimolar. Mr. Sandeep, under the guidance of Dr. A.K. Dubey, worked on drought tolerance studies in mango, and from his research work he concluded that two of the polyembryonic mango rootstock K5 and Kurakkan are drought tolerant. And if we are applying going for priming or treatment of the seedlings under drought stress with spermidine at the rate of 1 millimolar in case of susceptible polyembryonic varieties, and by salicylic acid 0.5 millimolar in case of tolerant, that may be very effective to enhance the drought resilience in mango rootstocks. This is all about the research. Now I will come. I am happy to share that our students are well placed, and last year 4 of our students, they qualified ARS examination and joined some ICAR research institutes. One of our students, Mr. Vishal Bahatre, earlier he was CTO and posted at Devang, but recently he got scientist B job in DRDO. Some of our students, they joined state departments, and very recently Mr. Bhupen Sagore joined IHR Bangalore as STO. Here again we are happy to see some of the recognitions or laurels our student brought for the division. Our Bhupen Sagore got UGC JRF Bush and DST Inspire Fellowship, Budding Scientist Award, and one of our students went for the international exposure visit, and several best oral and best post presentation awards are there. Publications, it is satisfactory for the year. Total 28 publication, out of which 13 is from national journals and 15 in international journal. The NAS rating is ranging between 5.53 to 12.63. In addition to this, some more publications are there. We could organize 2 training. One is supported by NAHAP CAST and another one is by DST Sir Karyashala. This is all about, I am really grateful to our Honorable Director Sir for all sort of support and guidance, and Dean Madam is there, Joint Director Research, Joint Director Extension, Head Division of Fruits and Horticulture Technology, Professor O.P. Avasthi. He is always ready to help the scientists and students, faculty, student discipline of fruit science, and heads of other discipline, those who are collaborating with our discipline, like post-harvest, plant physiology, biochemistry, our technical, administrative and supporting staff, and of course RA, SRF and young professional. Thank you very much, sir, for giving me this opportunity.
Okay, Dr. Manish, thank you very much.
Thanks.
for excellent compilation and presentation of your division's work. Thank you. Now I invite Dr. Ram Asri for presentation of management.
Honorable Chairperson of this session, but the Director and Vice Chancellor of ICAR, Dean Ma'am, Dr. Monica Joshi, Convener, Head of Divisions, Professors, Faculty Members, and my dear students. I will be presenting the significant educational achievement made in the discipline of post-harvest management. Sir, earlier this was post-harvest technology, but nowadays after BSMA guidelines, we are offering the degree in that post-harvest management of horticultural crops. So, moving to the strength of faculty, we have a very small numbers of faculty, that's 8, and coming to the student strength, presently we are having 9 MSc and 24 PhD student with the total number of 33. This is the mandate of the division where we are working. So basically the work of our student are in 3 thematic areas, that's the post-harvest management of fresh produce, processing horticultural crops and processing of arable crops. So these are the student MSc basically who will be receiving the degree in this convocation, Mr. Ajay Narola, Monica, Ranjani, Basla. Basla was conferred the best student and she won the merit medal this year. So coming to the work of MSc student, Mr. Ajay Narola worked on utilization of overripe banana powder for development of puffed extruded product. He chosen the overripe banana, made out the pulp and included different foaming agents and prepared the extruded snacks, and finding out of this was 7.5% egg aluminum could give the better extruded product which has a higher shelf life and nutritional quality parameters. Moving to the works of Monica, where she took the green pod cells, converted into pomace, and after that the stabilization of chlorophyll in this pomace is very difficult. So for that she stabilized the pH and used some gelating agent, that's a zinc, and finally she concluded that chlorophyll recovery can be enhanced by enzymatic assisted extraction method, and zinc is the best stabilizing agent, and addition of cellulose enzyme further increase the recovery of the chlorophyll from this pea pod based material. So Ranjini worked on persimmon, where she took the ripe persimmon fruits treated with the ultra assisted sonication and used the different sweetener agent, and among the sweetening agent she found the best one is xylitol. So with the use of xylitol she could prepare the best candy which has better functional quality parameters and storage life. Moving to the work of Basala V, as I told that she conferred this gold medal on her work day before yesterday. She worked on this ultrasound assisted extraction of pectin from citrus peel. So she extracted the pectin from citrus through UVA assisted extraction methods, purified it and worked out different functional parameters, and she concluded that the higher recovery, that is 26.23 of low methoxy protein. That's the beauty of this pectin. As the methylation is low, then this quality is higher, and the total phenolic contents were also found this in extracted pectin from the citrus, and this is non-toxic in nature and inhibits the lipid accumulation in hepatic G2 cell lines. That's a very useful in the pharma industry. So this is the list of PhD students who will be receiving the degree in this convocation. Mr. Ajit Kumar Singh, Menika M, Binod BR, Nirmal Kumar Meena, Vivek Shaurav, Bikono KSH and Vishnu Anand. They will be receiving their PhD degree in this convocation. Coming to their educational achievement, Mr. Ajit Kumar worked on post-harvest interventions for reduction of pesticide residue in apple, as the apple encounter maximum pesticide load. So he chosen the Royal Delicious apples treated with different pesticide, and after that he given the different sort of treatment, that's hot water treatment, gamma irradiation and oxalic acid. After that he extracted the material and calculated the load of this pesticide through LC-MS analysis, and he inferred that among the pesticides, dithion, malathion and dimethod are found the most susceptible to all the treatment on physical, chemical and the thermal treatment. The irradiation doses, that's in the range of 0.5 to 0.75 kg, were found most effective to neutralize the pesticide load in the apples. Moving to the work on Menika M, where she worked on guava fruits for enhancing the shelf life. She took the physiologically mature guava fruits treated with different doses of melatonin, brasinostride and UV irradiation, followed by storage at cold conditions, and she found that melatonin at the rate of 600 micromole per liter was found best, and coming to the UVC irradiation, that is found best in the dosage range of 2 and above, and that's ward of the anthracnose and watery soft disease in guava during storage. Anthracnose and watery soft rot disease are major problems during storage. Mr. Binod BR, he worked on papaya, and he treated the physiologically mature papaya fruits with UV radiation, ozonation and their combination. In salient finding, he found that UVC doses above 0.2 kilojoule was found to be best in preserving most of the desirable parameters and minimizing the anthracnose incidence during storage. Again, the ozone 2 mg per liter was found the best, and this ozone 2 mg per liter given the 5 days extra storage life in comparison to control during ambient storage. Another student, Mr. Nirmal Kumar Meena, worked on exogenous westernite application on mango fruits, and he treated the fruits with brasinostride, brasnozole and 1-methyl cyclopropane, stored and recorded the parameters during storage, and he found most these both two plant growth regulator, that's brasnozole and brasinostride, worked as a role reversal model. I mean to say, once they are used in higher concentration, they are going to suppress the ethylene production, and when they are used as at lower concentration, they are going to be triggered the ripening process. So this role reversal effect of these two biomolecules were observed in case of this study. Vivek Shaurav worked on this jackfruit pectin peel extraction. For that he collected the jackfruit pectin, extracted and purified, and characterized the different quality parameters, and he concluded that higher recovery of low methoxy pectin can be obtained with this extraction process, and the film prepared from the jackfruit pectin and tapioca starch and black carrot extraction has higher tensile strength. That's more, you can say, helpful in developing the edible packaging material. So that was the final conclusion of this study. Bikono cases, she worked on this capsaicin encapsulation with the different ball materials like cyclodextrin and this jackfruit pectin. So after coating this capsaicin with the different coating material, and she found that rice bran oil can be incorporated as a green co-solvent, and by tailoring extraction conditions, means combination of time, temperature, higher capsaicin recovery can be obtained. Release assess of microcapsule revealed sustained release of capsaicin and gamma original under simulated conditions. She did the simulation condition in vitro. So moving to another work, that's of Vishnu Anand, encapsulation of chia seed oil, and he took the chia seed oil encapsulated with the different wall material like tapioca starch, whey protein, maltodextrin and guar gum. And finally, he found that as the whey protein concentrate increased in the matrix ratio, encapsulation efficiency was increased, and increasing the oil loading in the microcapsule decreased the both capsulation efficiency and other parameters, reachable parameters. The maltodextrin whey protein concentrate was found as a ball material for encapsulating these chia seed oil. So these are the total 34 publications out of our students, total students, and they also published one practical manual and this 8 book chapters. So these are the details of the publications of our degree recipient. They published in very high top-notch journals, that's like Food Research International, having the impact factor like 14, Science Horticulture, Food Chemistry, so on and so forth, and these all the publications are non-paid publications, sir. These against the trail of other publications, they are mostly they are above 8 NAS factors. These are the some honor and award conferred on our students, notably this Bassala got the 50,000 cash prize award for her work, and she also won the gold medal. And these are the some honor award on faculty. I have been inducted as editor of Plant Growth Journal Regulation and guest editor of Act of Physiological Plantarum. I also received this fellowship from the Indian Horticulture Research Development Society. So these are some exposure visits made by our student at different places like Rohtak, Karnal and Sonipat during their study program. So with this, I profusely thanks to directors, dean, joint directors, head and faculty, student and technical staff, and all other members for giving the patience hearing. Thank you, sir.
Thank you, Dr. Sir, for your excellent presentation. Thank you very much for this important compilation.
Thank you, sir.
Now I invite Dr. R.K. Yadav for presenting the progress of vegetable crops.
Good morning to all of you, dignitaries on the dais, respected Director IRI, Chairman of the two sessions, Dr. Major Singh Sir, Dr. Monica Joshi, convener of the sessions, distinguished faculty members, students, ladies and gentlemen. I will be presenting the research findings of the MSc and PhD students of the disciplines of vegetable science who are going to receive degree during this communication. These are the mandate of the division, and we have the around 70 students on the roll, including MSc and PhD. These students include a student from the Assam also. Earlier, the student of the Jharkhand were also here, but now they are at their campus, and we have around 32 faculty, including the IRI, our regional stations and outreach centers. So we are going to present this research achievement under 4 themes, and there are 28 research theses we are going to present, 14 MSc theses and 14 PhD theses. And these are the list of the MSc degree recipients. They are the 9 from the IRI and 3 from the IRI Jharkhand and 3 from the IRI Assam also. We are going to present here in this session. So, under first theme, genetic diversity and nutrition studies, we are one thesis was conducted on this microgreen, that what are the vegetable-based microgreen, especially the effect of the genotypes, and photo-temporal studies were done by Ankita Saha, and they found that out of several genotypes of different crops, red cabbage and this red amaranth was found most suitable in terms of all the nutritional quality, and in terms of the light, they've given different types of lights also. So the blue light and red light were found very effective in enhancing the nutritional quality of these microgreens. And another thesis was this conducted by Mr. Ram Babu, and he was conducting his work on the gamma profiling, this gamma aminobutyric acid profiling in tomato, and he studied the regency protocol also. So he found that at green stage, at this gamma profiling was very high in different genotypes, and but in some of the genotypes this reduction at red stage and ripening stage was very less. That were the genotypes K63 and K70, and he studied the protocol for regeneration also in tomato. And another thesis was on characterization of this okra genotypes, and 79 genotypes were characterized in this, and this Pusa Bhindi 5 was found very high yielding and earliest in the flowering, and this through IPVS marker also the population was grouped into different cluster, and wild genotypes were also included in this study. So this was a very effective marker to distinguish the wild genotypes and cultivated genotypes. Another study was done by Barnali Majumdar. She was also in this study. She has done the gamma profiling of the genotypes, different set of genotypes which are suitable for protected conditions. So for protected conditions, she found that the orange color this genotypes, they were having at a very high GABA content, this at mature green stage, and ripening stage or even turning stage. When the ripening starts, and that time also some of the genotypes like DT1013 was found very high in GABA content. She also studied this in vitro regeneration protocol in tomato, and by taking different genotypes. And another thesis were conducted by Namita Rai. She studied the diversity in the garden pea, and she found that this Pusa 3 was found the earliest genotypes, and high protein content was found in GP102, and this was closely followed by Pusa Prabal, that was around 9% protein content. And another study was Ankit Kumar Sinha. He studied this diversity in pointed gourd. This study was conducted at IRI Jharkhand Center, and he found that HARP 79 was very promising genotypes, and he was very close to these all release varieties Swarna aulic, and the total of the diversity in yield contributing was the major parameters which contributed large in the total diversity of the genotypes. And they have done diversity analysis through this molecular markers also, and they identified one marker UBC 857 very high PIC value. And another study at also that IRI Jharkhand was conducted in okra. They studied the diversity in the okra genotypes there, and there also they found that the Pusa 25 was very high yielding per plant yield they reported, and Parvani Kanti was found a good combiner in this study, and some of the specific combiner also they found that they were very high yield and earliness in these different combinations like Pusa 25 and Pusa Makmali for fruit per plants. So they also they distinguish this characterize the genotypes with molecular markers, and different groups of clusters they form. Another Meghna Devi Reddy also she studied on this vegetable soybean, this nutritional profiling on the basis of the molecular morphological markers, and this biochemical profiling was also done in this month, and they found that AGS 458 that was a very good genotypes and having the basmati flavor, and this was very similar to Swarna Basundara, that another genotypes, and they could also find on the biochemical basis that linoleic and oleic this concentration of fatty acid was very high in vegetable soybean, that was at par to the this grain soybean. Another aspect was the breeding for biotic stress tolerance. So, in biotic stress tolerance for Kharif season, one study was conducted by Vahan Jenith, and he found that this Alternaria alternata was the major disease, and different isolate he found that this was the major disease. In this one, different isolate were very similar to that one, and some of the genotypes he found that this Nagina local and L9200 they were showing high level of the resistance to this Alternaria alternata, that is a purple blotch of onion, and they also found that in control measures that trichoderma, this ability was found better than other biocontrol agents for controlling this disease. Another thing, this we are also handling some under abiotic stress management, because this is a problem also nowadays, and under this one for drought stress conditions, cucumber, this physical parameters were and biochemical and this physiological parameter were studied, and it was found that relative water content, membrane stability index, these were the most of the parameters were very high in tolerant genotypes as compared to susceptible genotypes. On that basis, they could identify certain genotypes which were found resist tolerance to this drought condition, and on the basis of this heat shock protein, this expression of the certain heat shock protein was very high in that this tolerant genotype under heat stress condition as compared to susceptible. And this biochemical and molecular study of curding and flowering behavior was studied by Mr. Soham Luhana in case of cauliflower, and they found that this Indian type cauliflower, they were very tolerant to high temperature, and role of the GA3 was found very significant in the case of bolting, curding, and early seedling development stage. So these were the stages were very sensitive, and GA content was found very high, and on the basis of this expression of the flowering gene, they grouped the genotypes and found that some of the flowering gene were expression was very high in case of these genotypes. Another theme is the installation of the production system also, because some of the CPCT mostly we are working on this aspect. So in this one thesis was on this Kishore Barotia. He standardized these seed production techniques in Pusa purple broccoli one. That was the new release variety. So he has standardized and found that the 10 October planting at wider spacing of 60 into 45 centimeter, he got a very good quality of seed yield, and that was recommended by for production of the broccoli seed in the plain area. And another study was also done one in the Chile, where this net condition, because in the winter season, because it's very low temperature, we are unable to grow chili, and this one net condition was found very good for, and some of the genotypes like this CBL42 were found very good for this net condition growing, and some of the parameters like capsaicin, capsanthin, and ascorbic acid and other plant height etc. were very high under the net condition. Similarly, another study was done in lettuce by Thusal and in CPCT, and he found that some of the genotypes, especially the in lettuce genotypes, Lola, Roja, and this Great Lakes, they were recommended for very having the high nutritional quality, and they were in soil media and soilless media also they studied for growing of these genotypes. In PhD also, sir, we are going to present 14 theses, and 10 will be from II New Delhi and 4 theses from the IHR. So in this one, these are the list of the students who are going to complete their PhD, and these are the 14 students. And under genetic resource and diversity study, we found that one study was done on the ridge gourd, and in this one, this Lavanya, she found that some of the genotypes IHR, DMR, and IHR, DMR 18-65-2, they were found very good resistant to downy mildew in case of ridge gourd, and one marker SSR RG56 was found linked to this downy mildew resistance in case of ridge gourd. In breeding for biotic tolerance, most of the theses were in this section only, and this Santhia, she screened the most of the wild genotypes of the okra for she found that in that one that Evilmascus moschatus and Evilmascus tetraphilus were showing very high level of the YBMB resistance, and in the interspecific crosses, Evilmascus feculius was showing a very good crossability and seed set per percentage as compared to these two, and she did some this interspecific hybrid. She confirmed the hybridity with markers also, and two markers, this okra SSR21 and okra 56 were found very suitable for this distinguishing these genotypes. And another study was for yield related traits and mapping of downy mildew was done in cucumber, and they found that downy mildew resistant in our genotypes due to single recessive gene on the basis of this field condition and artificial screening they have found. And in case of this mapping, they found that the two QTLs on chromosome number 3 and 6 were correlated to the downy mildew resistance in case of cucumber. And another study was done in onion for stemphyllium blight, and they collected all the hot spot from hottest places, all the samples of this disease sample, and they found that there were very good similarity among all the onion races, and they were mostly the Stemphyllium vesicarum, and two samples from the garlic they collected, and that was the Stemphyllium eracnium. And they were also grouped the genotypes in different clusters, and they found that some of the genotypes also found some tolerance, like SB0113 and SB076, they were found tolerant to this disease. And another study was done for this watermelon bud necrosis virus in case of watermelon, and the population was studied, and the population was developed, and then they have on the chi-square basis they found that the disease is governed by single dominant gene in the genotypes, this DWM45, that is from Citrullus lanatus, and based on the BSA they found that one marker, that SSR marker, that was very linked to this resistant to this watermelon bud necrosis in watermelon. And another especially in the Balanehi at ICAR, she had done this mapping of anthracnose, and they have found that two isolates of this anthracnose were prominent there, and they had developed some marker, ITS marker from this one, and they screened the genotypes also, so they have initialized all this thing protocol, and they found that some of the in expression that 19 significant SNP also they were found resistant to this disease on chromosome number 2 and 6, and they also developed these CAPS and DCAPS marker from for this SNP. And another study was done on the mapping of a quantitative trace in watermelon for again this watermelon bud necrosis virus, that was done at IHR, and they found that VIL, they developed VIL lines, and 53 was found resistant to this watermelon bud necrosis virus, and they also found some closely linked marker because this was the VIL population, so very high level of this this PV they found in and QTLs they identified for this one. And another study by Saurabh Mahapatra was done in case of watermelon for gummy stem blight, because gummy stem blight is another disease which is very prominent nowadays, and they have found that IHR 617 and IHR 82 were found resistant to gummy stem blight, and they have gone for develop the QTLs for also, and very closely related QTL to this resistant to this gummy stem blight was identified in this study. And another study for this squash leaf called China virus is very prominent in the dry conditions, sir. And we have found that out of several viruses, this virus was very prominent, and our two genotypes, this DPU41, DPU43 showing resistant to this one, and resistant is governed by single dominant gene. In another study, this phylogenetic study was in the Luphi species, and 4 Luphi species were crossed, and it was found that very F1 very low level of this fertility was observed, and seed set was very poor, and especially the region for sterility was found that this absence of pollen viability, non-existence of anther, and abnormality in the meiosis abnormality. So on the basis of that, they found that this one cross, the cylindrica and gravulons, they were found that more number of this bivalent as compared to other crosses. So this was very closely related to this or cultivated type. And in another study, this Bhargav, he has done this interspecific hybridization okra and tetraphilus species and this moschata species. They found that in the one-way cross, that when this cultivated is used as a female, then it is successful, and while the reciprocal crosses was sterility was found very poor success was, and by colchicine treatment he can enhance the this pollen, this fertility of the F1, and he has also confirmed this F1 hybridity with the help of the markers and also flow cytometry method, and he studied the protocol of the regeneration protocol also, because the NOCRA protocol is not available. So far this will be helpful in pre-breeding material and other molecular studies. And another thing, the breeding for aquatic stress tolerance. In this one, in cucumber for heat tolerance, this study was done by Dhananjay, and he identified these different parameters for screening in this cucumber under heat tolerant condition. The chlorophyll and RWC, membrane stability index, they were found very high again in this case of tolerant genotypes. And certain genotypes, they have found that they are very showing high seedling stage and flowering stage also. And they have gone for this other stage like proline content and SOD, all these content were high in tolerant genotype as compared to susceptible genotypes. And they have found that heat shock proteins expression of the certain genes was very high in case of tolerant genotypes as compared to, and they found the QTL also associated with the heat resistant, and these were mostly on the chromosome number 2, 3, 6 and 7. And another study was done by in garden pea for heat stress tolerance, that there was grown in the early summer season, and they found that up to 39 degree temperature, and this 15 genotypes were so at least one pod or two pod you can get from this one. And they have clustered genotypes in different cluster also. And Aruna, he has done this mapping of a heat tolerance in capsicum, and he found that out of for 14 heat tolerant traits, 21 QTLs he found that were the major QTLs, and total QTL were 64, and 21 were the major one. And he studied the expression of heat shock protein also, and they found that 2 heat shock protein were expression was very high under heat stress condition. And these are the publications. This is basically only the research publication of the students, those who are going to get a degree in this program. So most of the publications were very good journals, and these are the list of some of the publications details. These are the awards received by our students because they participated in different seminars, symposia also. So they received different types of award, and these are the foreign visit of some of the students. They got chance to go on different countries and visit lab and their facilities to learn some techniques under this program. And these are the award to faculty also. And we have conducted one training under NAHEB, and Dr. Sawan Singh visited Japan under NAHEB to learn some techniques. And these are the PhD students, sir. Mostly they got jobs one or other places. MSc student also got some job. And last year 4 ARS were selected. And the state services, most of the students have gone in their respective state, like Karnataka, West Bengal. Most of the students, they are from that state. And we have conducted one exposure visit of this our students to the private sector also. Especially the Rasi seats we recently visited. So that was a very good experience for students to interact with private sector. So this type of exposure we will try to in future also with different areas. So these are the salient achievements of the division. Thank you, sir. And I thank all the persons for patiently hearing.
Thank you, Doctor. Thank you, Dr. Yadav, for very, very good presentation.
Thank you, sir. May I now request the chairperson for his kind remarks, please?
Dr. A.K. Singh Sahab, Director of this institute, Dr. Anupama, Dean, Monica ji, and all head of divisions, professors present in the audience. I'm really thankful to all of you for attending this important session and inviting me as a chairperson for this session. It was really nice to see the progress and the work done by the students in this organization. And the first presentation was Dr. K.P. Singh on floriculture, Manish on fruit crops, Ram Asreji on post-harvest management, then Yadavji presented on vegetable crops. It was all quality work has been done by the students. And it's really nice to see the interaction of quality faculty and quality students. Because last year I was interviewing the ARS candidates, more than 80% candidates from this organization only. And I was really surprised to see their performance. It was highly professional approach by the students of IRI, and very difficult to distinguish who is best, who is poor. All were almost equal. So it depends on how nicely you have trained them and nurtured them here. And even in selection also, I think more than 80% students got selected in ARS. Though we got many complaints also. The IRI students are surpassing. But it depends, because this organization is one of the best organizations which is nurturing best students, our future scientists. So I congratulate you. I don't have much time, sorry, because people are waiting for me. So I will end my introduction here. Thank you very much.
Thank you, sir. Director, sir, you would like to say something? May I now request Director, sir, and Dean, Madam, to kindly felicitate the chairperson?
Come to the conclusion of the session of School of Horticulture Sciences. Thank you, sir, for your kind words. And really, sir, we agree that we have a lot of quality scientists and quality students here. So, and thank you to the authorities and to the audience today. We have a good number of people in the auditorium. Thank you everyone to attend, and we will break for tea now, 15 minutes, and then we'll reassemble, please. Thank you.
Oh, shit. चलो रह गया हाँ? and warm welcome for the second session of the day. Myself, Dr. Monica Joshi, Professor in the discipline of Seed Science and Technology and convener for 2 days professor presentation, along with my co-convener Dr. Shruti Sethi, Principal Scientist, Food Science and Post-Harvest Technology, heartily welcome all the esteemed dignitaries, faculties and students to the 6th and last session of the professor presentation. The chairperson for today's second session on social science, Dr. V.V. Sadamate, former advisor Agriculture, Planning Commission, is amongst us today. Sir, it's an honor for us to have you here. May I now request Director Sir to kindly welcome the chairperson with a bouquet of flowers. I now invite respected Dean Madam for the welcome address.
Thank you, Dr. Monica. A very warm welcome to all of you, especially my dear students, to this last session of the Professor's Presentation Program, which was a 2-day event, sir. And I formally welcome Dr. Sadamte ji on this very important, for this very important session. And thank you very much for agreeing to chair this session. And we always solicit your advice regarding the research and the extension outcomes of the social sciences program. So we have here, sir, in this session, the professors of the 5 disciplines: agricultural economics, agricultural extension, agricultural statistics, bioinformatics, and computer application, who will be making the presentation on the significant educational achievements based on a total of 73 research theses comprising 32 master's and 41 PhD research thesis. And here, as usual, Dr. Monica Joshi and Dr. Shruti Sethi, they will continue to be the convener and co-convener. Now, without wasting more time, I request Director IRI to kindly apprise the audience about chairperson of today's session. Thank you very much.
So welcome once again to this second session on social sciences. To chair the session, we have with us Professor Dr. V.V. Sadamate, former advisor, Agriculture, Planning Commission, Government of India. And he is one of the most distinguished alumni of ICAR. He did his master's and PhD both in agricultural extension from ICAR, New Delhi, 1975 and 79 respectively. Worked with Ministry of Agriculture as director, and then as additional commissioner extension. And further he joined as Planning Commission as advisor agriculture. And it was a long stint that he worked with Planning Commission from 2006 to 13. And he was instrumental for preparing the 11th and 12th plan of agriculture and allied sector. So, and that has given, in fact, boost to our GDP as far as agriculture is concerned. He has contributed significantly in developing operating, extension, innovation and reforms. And he is associated with IRI as adjunct faculty since 2014. And he has been, of course, associated with several agencies which have been involved in formulating the policy framework, guidelines related to extension. Currently, he serves as a freelancer extension specialist. Anybody who has any help required in the field of technology transfer, because you know that extension is one of the most fast developing discipline these days. The digital system of extension is picking up so fast that the conventional method of extension is almost getting obsolete. And if we don't pick up with the technology of extension, which private sector is doing very efficiently these days, weed seed company, weed fertilizer company, pesticide company, they have taken up this upfront. And that's where it becomes all the more important that we evolve in our extension methodology, and that's where Dr. Sadamate is playing a very important role Just now he was talking about, having an international meeting in coming March where a extension system and platform, at least for Southeast Asian countries, where the situations and requirements are almost similar of agriculture can be developed, and therefore the countries can benefit from one another's experience. So he is deeply involved into that, and he is a great well-wisher of IRI. Anything that, whenever anything comes to IRI, his heart and soul is always there. Thank you, sir, very much for accepting our invitation and being here with us. Thank you so much.
Thank you, sir. So as convener of the session and with the permission of the chair, I would wish to apprise the House regarding the modalities for the sixth session. For the sixth session, sir, we have 5 presentations in the following order. First, agricultural economics, to be followed by agricultural extension, then agricultural statistics, bioinformatics, and computer application. For all the 5 disciplines, sir, we have already given the allocated time to all the professors. And like for yesterday and for today morning, kindly highlight only the significant research outcomes without going into further details. Before the finish of the allocated time, 2 minutes prior, the yellow light will blink, and when the time is over, the red light will blink, so that you can accordingly wrap up your presentation. So with this, I now hand over the session to respected chairperson for conducting the further proceedings. Thank you.
Good morning, everybody. Honorable, Honorable Director Sir, Dean Madam, Madam Monica, senior faculty members, presenters, and students. The format is explained to you by Madam Monica. And sir, I'm friend of all these presenters, but when it comes to time management, I'm a little, kind of non-friendly, because time is very short. And as Madam Monica had mentioned, just go for the highlights of significant studies. Otherwise, if we try to listen to all of your presentations, then there are 73 studies covering agriculture economics about 16, then agriculture extension 20, then agriculture statistics 15, bioinformatics 14, and computer application 8. So all 73, if you go into the details, then perhaps, we may not be able to, catch up with the time. So as, coordinator had very rightly mentioned that each one of you have been given 15 minutes approximately, and computer application gets approximately, 10 minutes. Try to, manage within that. And for a learned audience, perhaps the findings and implications will be more important. But at the same time, there is a study which has done very, very in terms of innovations in methodology, innovations in analytics. Bring it out. Time constraint apart, if there is any significant point which you would like to submit to the audience, then we can extend for 1 or 2 minutes if it is that manner.
Sir, may I submit in this context that there is already a pre-information communicated to the professors, and they have been allotted some 20 minutes time, and in that a format has been given, and I'm sure the way since yesterday sessions are going, sir, means they are taking lot of care on that. Yeah.
Right.
So, sir,
So we begin with Pramodji.
Most respected Sudhamte sir,
Green light again.
Dr. A.K. Singh sir, Madam Anupma, Joshi Madam, and Dr. Padaria sir, Brahmananda sir, faculty members, and students. I'm going to present the significant educational achievements for the Division of Agriculture Economics. So we have this year, as stated, 16 students are taking degree, and we have published number of papers, and so the major themes and are the technology, resource, and environment, and under this 6 students are taking the degree. And then the next theme is the agriculture market and value chain. In this also 6 students are taking degree. Then the third one is the institutional innovation and rural livelihood, in which 4 students are taking degree. The first one is the study is the economic assessment of human-wildlife conflict in agriculture. As we know that it is a growing concern, and therefore this study was taken up by Naveen Kumar Nayak to understand as to how much is the loss he wanted to estimate, and what mitigation measures could be taken up. And we find that the economic loss ranges from 23,000 to 34,000 rupees per farmer. And he also tried to estimate as to what is the extent of loss in terms of the crop loss is concerned, and the proportion of loss ranges from 9% for, it varies from crop to crop based on the nature of the crop, and the average loss is 2,000. It also, the loss was estimated to be 2,000 rupees for horse gram, and it went up to 43,000 for groundnut. So it varies, this was the estimate which was made, and then he also tried to estimate as to what is the willingness to pay by the farmers through a detailed contingency valuation method, and he estimated that the farmers are ready to pay up to 508 rupees per acre, and he was trying to devise a mechanism by which this could be included in the Prime Minister Fasal Bima Yojana, so that the crops are insured and the farmers are, the risk is mitigated. The next study is on the climate change, vulnerability and risk management in agriculture. In Maharashtra, this was done by Athare Prakash, and he created a index consisting of the exposure index, sensitivity index, and adaptive capacity index. And then using all these 4 measures, he devised a composite index and categorized the districts into high, medium, and low vulnerable districts. And measures were suggested as to how we can mitigate in these high vulnerable districts. And he identified that the Marathwada and western part of Vidarbha region were more vulnerable to climate change. And he also estimated the impact of climate variables on crop yield, and it was found that the increase in maximum temperature had a negative effect on the crop yield. And farmers are mainly exposed to drought risk and insect pest risk, and therefore he suggested that the crop planning needs to be adjusted according to the climatic conditions, and implement adoption and mitigation measures by educating farmers through training, exposure visits, and awareness programs. The third study, which was on conjunctive use of farm pond and groundwater. We know that the farm ponds are very important, but in Karnataka, what is also prevalent is, along with the farm pond, groundwater is also being utilized. And the conjunctive use, we wanted to know as to how beneficial these are, and whether it is beneficial or not. So this study estimated that the external benefits of farm ponds are to the tune of 89,000 rupees, and the external cost are 80,000 rupees. And one of the hallmark of this study is the social cost benefit analysis was done. Usually we do normal benefit cost analysis. This case, since the farm ponds and the groundwater both have the social cost and benefits associated with them, so that was taken into consideration for estimating the benefit cost ratio. It was found that it is feasible to, the conjunctive use of farm pond and groundwater is feasible,
He also used the propensity score matching to understand how much is the gain in terms of the yield, and it was found that the farm ponds over rainfall resulted in a significant increase in yield. Another study was on an economic analysis of food systems and food security in Sierra Leone. Nabi is from Sierra Leone, which is the western part of the Africa and continent, and he tried to estimate as to what is the food security seen in the country, and what he found that food insecurity is to the tune of 64% of the population is suffering from food insecurity, and the Sierra Leone faces food challenges, but promoting diversity and supporting farmers offers hope. The interconnectedness of food diversity, food security, and malnutrition in Sierra Leone emphasizes the need for comprehensive interventions. And he also analyzed as to what is what he can take from the country, from India, and he also studied the food policy of the India, and he understood that, and based on that he suggested, the study suggested that to develop public distribution system leveraging technology for efficient and timely delivery of food grains, addressing leakages and ensuring social safety nets. So another study was on estimating the economic and environmental benefits of direct seeded rice. We all know that we are also promoting direct seeded rice, but there are certain challenges with this technology, and we wanted to understand what is how much is the gain as a result of adoption of direct seeded rice. What are how we can overcome the challenges. The adoption of DSR was highest in, it varies across districts in the Haryana state, and Haryana cultivated DSR to the tune of 1.93% of total rice cultivated. And it was estimated that the DSR leads to reduction in the yield, and this DSR is being promoted because of its environmental benefits, and we try to estimate what is the environmental gain as a result of this adoption of DSR, and the value of carbon saving due to DSR was estimated to be 7,419. And he also suggests, the study is suggesting that we must incentivize the farmers to the tune of 7,933 in order to promote this technology. Then another study is the institutional and technology for enhancing efficiency and equity of groundwater irrigation in India. So groundwater is very important, especially in the western and southern part of the country. What we find that the groundwater levels in southern and western parts have shown improvements, while the northwestern and desert regions continue to be hotspots because of excessive use of groundwater. Then the agricultural electricity subsidies positively contribute to agriculture production, but it also leads to greater use of groundwater. So that is one challenge. So the efficiency of farms in some humid regions were found to be higher than those in the arid regions. So we try to estimate the technical efficiency. The micro irrigation has led to reduction in groundwater use because but the amount of groundwater use has increased. So that is one challenge, and therefore the study emphasizes that need to enhance the use of efficient groundwater management strategies. We also need to design location-specific policies. We also need to educate the farmers so that, and also have policies and regulations for raising the marginal cost of water, and so that, the efficient utilization of water is there by the farmers. Another study which we took up was the impact of blending of edible vegetable oils on rapeseed and mustard. So blending was allowed in the year 1990. So the blending of oils were allowed in the year 1990, and it was stopped in the year 2020. So we wanted to estimate what is its impact, because once the blending is allowed, that affects the industry as also the farmers, because the farmers, they do not realize a better price, and therefore what are its implications we wanted to know. And the study found that the growth rate of area production and yield was high during the before the blending was there, and so that is shows one of the impact of blending. And we also tried to know as to what are the factors responsible for acreage allocation in rapeseed and mustard. And the previous year's acreage of rapeseed and mustard has a significant effect on the acreage during the coming year. And as far as the impact is concerned, we had, the blending was stopped in the year 2020, and after that, we had 3 years period when the blending was stopped, and therefore we forecasted using the NNAR method as to what will be the, the area production and prices during these 3 years, and the difference in the actual and the forecasted area production and prices gave us as to what is the could be the impact of the blending, and we find that it is to the tune of 52% increase in area was observed as a result of stoppage of blending. Then we also the division took up the study of performance analysis of ENAM and its impact on price realization in Rajasthan and Haryana. ENAM is promoted as one of the strategies for providing better market access to the farmers. And what we observed that the ENAM trade has increased more than 17 times in the last 5 years. So the trade volume is increasing, and its reach is, to the farmers is also increasing. And we also observed that if we see that the farmers are ENAM versus the AgMark net prices, if we analyzed, and it was observed that the prices prevailing in ENAM, what the farmers are getting in ENAM is more than what is received through the AgMark net, and therefore this is beneficial and needs to be further promoted. We also assess the awareness level of the farmers and the constraints, and one of the constraining factor was the account payment is known to the farmers, but acceptability is less. So the farmers, they prefer that the payment should be received in hand or in cash or physically to them. Farmers also felt the need for their presence during the auction period, because this ENAM feature is that the auction takes place and the trading takes place without physically the different entities being there. Then another study, important study we took up was, the division took up was competitiveness, concentration and capacity utilization of oilseeds processing industry. So this is one segment, one of the key stakeholder of the value chain, oilseed value chain, and that also needs to be understood as to what is happening at the oilseed processing industry, how they're performing, and so there what we find is that the industry is stagnant as far as the number of units are concerned, though the volume, the output is increasing. And as far as the composition of the industry is concerned, we found that the vegetable oil and fat manufacturer, that they constitute the largest number of the industry, followed by the hydrogenated oil and vanaspati ghee manufacturers and others. The share of the oilseed processing industry is more in the was observed more in the urban regions, and it is slowly taking the urban shape, moving away from the rural areas. And we also assessed the net protection coefficient, which was found to be more than 1, revealing that the competitive disadvantage is there as far as the oils are concerned, as also the oilseed. So we need to be more competitive if we want to reach out to the international market. And the study employed the Herfindahl-Hirschman index to assess what is the how the market structure is there of the oilseed processing industry, and there we find that it is becoming more and more competitive, and more number of players are coming, and that is how the competitiveness is increasing, which is a good sign for any industry. We also assessed, the division also assessed the multi-stakeholder analysis of e-marketing of agricultural commodities, and so this is another study in the area of the e-NAM, we can say. And through primary data, we try to assess and analyze, and it was observed that the e-NAM covers 13.4% of the farmers of India, and Uttar Pradesh stands top, followed by Madhya Pradesh. So these are the two states which are performing very well as far as the e-NAM is concerned. E-marketing participants realize higher gross return to the tune of 3,902 rupees per hectare, and they also realize a better price. So this confirms the previous study, the findings of the previous study. They are almost in the same line. And farming experience, farm size, and access to extension services had a positive effect on the farmers' participation in the e-marketing. So these need to be promoted in order to bring in more and more farmers under the to use this e-NAM framework for marketing their products. This was a very good study, which was developing a multi-market and multi-regional analysis of important cereals was done. This was presented for the PhD program, and this is a very good model which was developed and can be utilized for various other commodities in order to forecast as to what will happen to various policy shocks as far as the production or productivity or trade of important commodities are concerned. Impact of FTA, foreign trade, free trade was analyzed as to what will happen if the free trade is there between the two countries, and this was a smart model was used for this to analyze, and it was found that
Pramod ji,
trade.
आपने yellow देख लिया ना?
Yes, sir.
You've seen it, huh?
Yes, sir. Thank you, sir. So, economic evaluation of seed production of was assessed, and IRA has 4 kinds of model which was used for the study, and it suggested that the IRA SPU model was the most efficient model, which was giving higher prices to the farmers. The another study was determinants of performance of farmers producer organization, and the next was on the determinants of performance of agribusiness startups in India, and startups is growing at the rate of 32%, and some of the states which are dominating in the startup, and there are 3 critical factors which promote FPOs. Then changing, this is the last study, sir, changing rural employment pattern and its implication on farm economy. We find that the wage rates are showing a slow growth in recent years. The real wage of male is greater than that of male, and rural workforce is undergoing a structural transformation from agriculture to non-agriculture sector. And we have this these 3 training programs were organized in the division, and some of the important publications, students' publication, the faculty publication are equally good, and the recognition by the students, and faculty awards and honors, and finally, thank you, sir.
Thank you, Pramod ji. Monica ji says that we will have presentations first, and then clarification, interactions later. After all the 5 presentations. Thank you, Pramod ji. Thank you very much. We'll come back to you on observations.
So next week agriculture
Nainsa, please take over.
Respected Chairman, sir,
Honorable Director, Dean Madam, PDWTC, Director ICAR, professors, heads, respected convener of the session, co-convener, students, ladies and gentlemen. A very good morning. In agricultural extension education, we have 23 faculty members. Out of them 13 are research guide. During the year we had 79 students, including from IRI Assam, IRI Jharkhand and Raipur hubs. Out of them 20 are receiving degree this year. We have 6 MSc students who will be receiving degree who worked on 4 different theme areas. Ms. Sahiti worked on evaluation of mango value chain of farmer producer organization. She presented her presentation in gold medal for a master's. Detailed presentation was given. Through Porter value chain, she identified some of the areas for cost minimization, and there were 8 marketing channels she could identify, in which the farmer marketing channel with export to foreign national received the highest price for all of the channel. There was almost 38% increase in productivity and 22% increase in price received per ton by the members in comparison to non-members, and there was overall output value of members was 68% higher. 48% women were empowered through participation in FPOs, but their involvement was very restricted. 17% had felt that they had gender parity, and she identified some success factors for success of FPOs. Another study was done by Mr. Anirban Jana, where he tried to evaluate value chain of IRI wheat varieties, and he could found out that in northwestern plain zone 4 value chains he identified, in which farmers, FPO, government agency was the most popular one with the highest marketing efficiency, whereas in central zone the farmer, commission agent, miller, wholesaler, retailer to consumer was most popular one. In both northwestern plain zone as well as central zone, the combination of grain yield more than 50 quintal, bold grain size, market rate higher than non-IRI varieties, dwarf plant height, less pest and disease infestation, crop duration nearly to 110 days and 3 to 4 irrigation with a straw yield of 40 to 50 quintal per hectare received highest preference. Whereas while comparing average cost of cultivation, it was found out almost that 38,000 per acre, and average net farm income from farmer per farm was almost 11,000 per acre, and he also calculated the ratio of input to output. Another, in the second theme, assessment of government centric programs, Ms. Mathi Grishma worked on access to extension and advisory services, and she found out that almost 49% were accessing extension advisory services. Progressive farmer were topping, whereas in northeastern region, Mizoram accessed highest extension advisory services, age, education level, irrigation, training attended, operational land holding, membership in farmers organization. They were the significantly and positively determinants of extension advisory services. Whereas gender, household size, social category of the farmer were negatively affecting the EAS. Women had more access to extension advisory services in fisheries sector. Non-farm income played more role in the agriculture for increasing access to extension advisory services. Mass media and private sources were prominent role player in increasing the productivity, whereas information access increased with an increase in size of land. Small and marginal farmer relied more on progressive farmer and private extension input like input dealer as source of extension advisory services. In another study, the public distribution system was analyzed in Bihar, by Amit Sinha and found out the significant difference between the perception of public distribution system beneficiary and non-beneficiaries. The beneficiary having medium knowledge regarding PDS and nutrition was significantly different than non-beneficiary farmers. The amount of food reaches to fair price shop was lower than what it was needed, and corruption at all levels of food distribution to fair price shop was perceived as major constraint in implementation of public distribution system. Whereas malpractices in weighing, insufficient quantity, non-timeliness of food supply and unsatisfactory quality were major constraint in availing these services. Another student, Mr. Naveen Kumar, worked on media and ICT, whereas he tried to assess the social media effectiveness in increasing the adoption of finger millet, and found out that farmers' social media usage behavior, including their experience in social media, regularity of use, amount of time spent, usefulness of social media and utilization of social media were the determinants. Low knowledge on services available in social media, lack of authenticity of individual in social media, consumes more time to get information, and poor and late replies by the expert were perceived as the major constraint. Mr. Leela Krishna Chaitanya worked on health and nutrition issue and found out some postural agronomic assessment in paddy cultivation in coastal region, and he established that among work-related health hazard, pain in body, fatigue and numbness in the hand were the most prevalent among male and female workers. The male-dominated activities like energy expenditure rate, cardiac cost of recovery, cardiac cost of work, physiological cost of work, total cardiac cost of work were high in threshing and harvesting activity among female-dominated activity. So Rula, he tried to establish that farm workers were at very high risk in harvesting, threshing and transplanting activities, whereas through Riba, he established that farm workers were at high risk in load carrying activity. Mean rank for adoption intention of drum seeder among all the improved tool was found to be very high. Among the PhD degree recipient this year, we are receiving 14 students are receiving degree who worked on 6 different thematic areas, and in first theme, assessment of government schemes, first study was conducted by Mr. Praveen. He worked on the Operation Greens, a government program, whereas he tried to establish that there were certain constraints in top value chain development, tomato, onion, potato, the lack of proper utilization of processing facility, delay in payment, lack of quality produce, high price fluctuation, lack of participation by the project members, and he established that farm FPOs in the top crops are working for increased crop productivity, enhancing the marketing and negotiating skill of the farmer, value addition through processing and contract farming. He used a method named Demetal, through which he tried to find out that farmer socio-psychological factor, market intelligence for top crops, input supply under Operation Grain was the effect of inclusion in FPOs, whereas government policies, support from grassroots level extension were the causes of the output. He tried to find out the comparative effectiveness of the project sanctioned under top, and almost in all dimensions Wangi Foods was topping among the 3. Another study by Ms. Chiru Grace was conducted on gender evaluation of integrated farming system in Manipur, where she tried to find out the knowledge of the respondent, and there was significant difference in the knowledge of male and female adopters of farming system. The economic constraint appeared to be the most significant and challenging for both the gender. Socio-personal, technical, situational and other marketing constraint also played the role. The another thematic area in nutrition and health was taken up by Mr. Chennaveres, and he tried to find out the awareness and knowledge and established that 23%, 42% and 36% respondent for pearl millet belong to the low, average and high category, and 25, 43 and 32% respondent for rice belong to low, average and high category respectively. He tried to find out various strategy for adoption of biofortified crops, and perceived behavior control was found to be most influencing for the intention to adopt nutri farming. Farmers need to be given training in adoption of cultivation with awareness, since subjective norm was playing important role in then capacity building of farmer was to be advocated, and he also tried to find out intense promotional campaign and supply chain subsidized seed, and they were the factors through which we can increase the participation of farmer in biofortified crops. Effectiveness of public distribution system was assessed by Mr. Surjakant Roy for nutritional security, and he established that the Nutri-PDS effectiveness index was having accessibility, degree of contentment and cost saving as the three more most effective dimension, and Tikamgarh district was found to rank first, followed by Chitrakoot, Chhatarpur and Jhansi district. He also tried to establish relative importance of Nutri-PDS effectiveness index component and find out some factors ranging from E1 to C2. Through discrete choice experiment, he established that pearl millet being the most preferred, followed by the sorghum, preference for nutri-cereal as additional choice to substitute the rice, and there was no clear cut preference from wheat to the nutri-cereals. Then Ms. Sai Priyanka worked on identification and validation of indicators for the model agri-nutri smart village. She worked in Uttar Pradesh and Telangana, and they tried to compare both the states and adopt agri-nutri smart village versus non-smart villages. The farmers in Telangana fell moderately knowledgeable, whereas in Uttar Pradesh they were most knowledgeable. The attitude scale identified 5 key factors. In agri-nutri practices, majority of the farmer in Telangana and Uttar Pradesh were under medium category. Among all knowledge, attitude, perception variable, the respondent from model villages of Uttar Pradesh and Telangana performed well than non-model villages. The result of structural equation modeling showed that relationship between knowledge and attitude, knowledge and practices, and attitude and practices were statistically significant. She also established through PCA 12 factors starting from production diversity to sustainability. In the results, she identified that 77% of farmers in Telangana model village were agri-nutri smart, whereas 72% farmer of Uttar Pradesh were agri-nutri smart. Nutrition and learning was followed by smart technology dimensions. The results from ANAVA showed that significant difference between model village and non-model village of both the states, but with no difference among model villages across 2 states. The positive significant association of index score confirmed the sensitivity of index with not much difference attached to weightage procedure. Cues for action, self-efficacy were major determining factors among the model villages versus non-model villages. Among health belief model factors like perceived severity, perceived benefit, perceived barrier were more significant. Among socio-economic factors, the age, education, land holding, annual income, food expenditure and scientific orientation were associated. She also advocated some strategies for it. In the next thematic area, entrepreneurship development, Mr. Ashish Kumar worked on analytical study in linkages of selected enterprises, and he selected crop-based enterprises and two dairy-based enterprises. Through network structure and analysis, he established that farmer of all the enterprises signified the importance of strong ties with families, neighbors in agriculture, even when they had connections to external researcher and extension agents. Weak ties were established as to be important to introduce innovations and new learnings, whereas strong ties were highlighted for adoption of innovation by a larger group. He established the farmer's attribute with adoption and established that Atma and KVK had greater degree centrality, indicating more direct connections. State government agricultural official had higher betweenness centrality, more control over the flow of information and resources. Input dealer were found to have similar eigenvalue centrality with farmers of respective region. Village traders had higher closeness centrality, which refers that it is more closely connected with one another. Whereas in the determinants, age, sex, education, income, attribute had significant relationship with centrality measures. In another thematic area, innovations in extension system, Mr. Sunil Kumar worked with the community managed extension system for sustainable agriculture, a multidimensional study, and in Maharashtra and Andhra Pradesh he tried to find out various factors related to ZBNF. He was able to find out that farmers became 13% more likely to use new varieties and soil preparation method, 15% to proper use of natural fertilizer, and 11% to use the manure and compost. Whereas education, farm participation, extension, contact, integrated soil fertility management practices, drip irrigation, use of waste decomposer were major determinants of adoption of sustainable agricultural practices through ZBNF. Through social network analysis, he established that ZBNF trained farmers had the highest degree centrality, indicating its significance as most preferred information sources on ZBNF related information. Progressive farmers, cooperative and NGOs in Maharashtra, cooperative and NGOs in Telangana were also associated with ZBNF trained farmers determining factors. ZBNF was able to produce some strategies were advocated by the researcher that ZBNF produce should be certified before going to value chain, setting village level input preparation and more number of farm demonstration in addition to subsidy and having some lower side establishment of new
3 minutes more. 3 minutes more.
Or then an analytical study on livelihood and climate change adaptation in biospheres was conducted by Ms. Aishwarya. She presented for the gold medal also, and she established that human wildlife conflicts, forest fire, monoculture plantations were some of the socio-ecological concerns. Adjustment in crop calendar and crop diversification were among the crop-based, livestock-based, fisheries-based and other adaptation strategies. The communities depending on forest-based strategy were food insecure in comparison to business-based and crop-based. Some of the determining factors were also established. The community judgment on design and content of a learning E-module was found that the e-learning module which she designed was very applicable, comprehensible, and it was associated with problem solving. She established that a major concern for success factor governing the effective management of biosphere reserve can be equity, efficiency, ecological sustainability, decentralization, interest, and incentives. Another thematic area, media and ICT, Mr. Yagnesh worked on utilization of ICT in National Agricultural Research System for enhanced efficiency through structural equation modeling and another analysis. He established that there is clear strength in strategy, technology, and human resource support. Required improvement were the organizational support aspects of e-culture readiness, strategic initiative, the system quality, information quality, and service quality exerts a significant influence on system uses, and in turn system uses significantly impact the net profit. He tried to extend the technology acceptance model by incorporating perceived effectiveness and organizational support, training, and computer self-efficacy emerged as the significant drivers. Coefficient of determination through the squared multiple correlation for system uses and user satisfaction showed substantial variance accounted by external variables. The constraints were listed, and multiple login, lack of training, multiple entries were the major constraint. Ms. Sonali Malik tried to find out the effectiveness of Pusa Samachar, a multimedia based extension model of IRI for its information delivery and uses. When she established information need and found out that 55% farmers need information on rice, 55% need information on wheat, whereas 53% need information on using multimedia. Information seeking behavior using multimedia was significantly determined positively by personal cosmopolite context and personal localite context. Degree of innovativeness significantly determined the information need on rice. She found out the 6-factor model to measure farmer behavior intention to use multimedia based agro advisory services. The 6 factors like trust to our source, self-efficacy, effort, expectancy of farmer, they were having positive and significant effect on perceived benefit. About perception of the quality of Pusa Samachar, 71% respondent had neutral perception towards it. In another study, Ms. Gangubai tried to find out effectiveness of agricultural programs of Doordarshan, and through stepwise regression analysis, she established some of the facts, and utility score was found in the first row, whereas horticultural programs with crop protection as the content and was the first one. In contemporary issue, innovation for adoption of climate change and natural disaster was attempted by Mr. Sudeep Gorai, and he established that livelihood strategies, income, food access were enhancing resilience. He also developed and validated a e-learning module on farmers' resilience to climate change induced natural disaster. Ms. Preeti Yadav worked on farmer-led conservation strategies, improvement of crop genetic resources, and she identified 30 paddy land races, and through her analysis in Western Odisha and Eastern Odisha, Uttarakhand, she find out some indigenous practices, strategies, and established that value belief norm model holds good, and she also developed a model, e-learning model, which was rated very good by the experts. Last study was conducted by Ms. Susmita Seni. She tried to understand the migration behavior of agricultural household and establish some relationship through analytics that the migration of household male member lead to more engagement of women in making decision related to agricultural. She found out some alternative rural employment opportunities and established a specific threshold for farmer and a monthly income of 28,000. He or she should aim for 1600 liter of milk per animal, rabi yield of at least 22 quintal per hectare and kharif yield of 16 quintal per hectare to reach this income target. We had 45 research publications during the year, 2 training manuals, 14 conference, seminar, souvenir presentations, 13 book chapters. The 29 out of them are from student side and 16 publications are from faculty side. We had the recognition of student in various seminars, symposia. 5 students of this division got selected in ARS, 2 assistant professor, 2 SMS in KVKs, and our faculty member were equally recognized. We conducted 2 training courses during the year, and division is providing the services to the students from SAUs, farmers, visitors through its visitor cell, and almost 18,000 visitors were entertained during the year. We are also offering 2 courses to the whole of the institute, PGS 505, 502. With that, I sincerely thank Honorable Director, Dean and Joint Director, Head of the Division, faculty members, students, respondent of various studies and the graduate school for sincere support. Thank you.
Thank you, Dr. Naik. Thank you very much, sir. We'll have a couple of questions and interactions.
Thank you.
Maybe a little later. Huh? Thank you very much, sir. Now, Sini Madam.
Educational achievements pertaining to the discipline of agricultural statistics located at ICAR ISRI. Statistics and numbers are no good unless you have good people to analyze and then interpret their meaning and importance. At present, we have 32 faculty members. Out of them, 3 are also faculty members of bioinformatics discipline. 15 of them are eligible for guidance. We are offering MSc and PhD program in agricultural statistics. 28 and 19 courses are offered in PhD and MSc respectively. Our faculty members are also involved in 10 UG courses at IRI, and we also conduct senior certificate courses in statistics. Our first batch of MSc came out in 1966 and PhD in 1968. We have a proud alumni group of 383 and 228 in MSc and PhD respectively. At present, we have 14 and 37 students in MSc and PhD enrolled students now. These are the list of degree recipients PhD this year, along with their guides. There are 7 students in PhD and there are 8 MSc students. They are shown here along with their respective research guides. Our discipline major areas we can divide into 4. 5 students each worked on design of experiments and statistical modeling, 4 on sample surveys and 1 in genetics. First theme area is design of experiments. Our PhD student Vinayaka worked on PBIB designs based on higher association schemes and their applications. He has obtained some efficient classes of resolvable, minimally replicated PBIB designs, nested partially balanced treatment incomplete block designs and nested partially balanced bipartite block designs. The designs developed are very efficient, and they, when there is a resource crunch, they are especially useful. Resolvable designs are useful particularly in sequential experimentations. The bipartite designs are useful for test treatment versus control comparisons in biological studies where one factor causing heterogeneity is nested within the other. Another PhD student, Ankita, worked on some aspects of asymmetrical response surface designs. She has obtained asymmetric third order rotatable designs that has reduced run sizes, 3 series of designs for fitting second and third order response surface models for asymmetric factors involving quantitative as well as qualitative factors were obtained. So in summary, her output is efficient sequential response surface designs for symmetric as well as asymmetric factors, which are best in terms of cost, predicted response precision and nearly orthogonal parameter estimation. Our third PhD student, Neha, worked on experimental designs involving doubly nested blocking factors. These designs are also known as tri-hierarchical incomplete block design. She has obtained 3 methods of developing such designs and 1 R package. These designs have very small sub block size and can be directly used in obtaining designs for breeding trials. So she has developed 4 methods of construction of mating environmental designs involving diallel, triallel and tetraallele crosses using these designs. A comparative study of these designs for unequal block sizes versus equal block sizes using mixed effects model was also taken up by her, and it was found that with equal block sizes they performed better. Our MSc student obtained some series of cyclic minimal balanced and minimal strongly balanced crossover designs. These designs are especially useful when the treatments are to be applied in succession without any washout period, and there will be an evidence of residual effects. So he has compared his designs with the corresponding row column designs and found that these designs are quite efficient, and both methods of constructions were implemented using R. Another MSc student, Rakesh, worked on algorithmic approach for construction of uniform designs. 2 new algorithms for constructing uniform designs in rectangular and irregular regions were obtained. The first algorithm addresses the deficiency of existing methods for rectangular area. The second algorithm focuses on construction of uniform designs on irregular regions, demonstrating improved efficiency of these designs. And he has also developed R codes for the easy implementation of those algorithms. These are some of the publications by the design group of students, and they have developed 2 R packages which are well received by the researchers across the globe. These are the conference presentations by the students in design group. Now coming to the second theme area, that is sampling techniques. Our PhD student Ashish worked on development of geographically weighted regression estimator for multicollinear survey data. His findings include ridge regression and least absolute shrinkage and selection operator have been used to nullify the effect of multicollinearity in complex survey data. GW ridge and GW lasso estimators of population total were developed in presence of multicollinearity. Extensive simulation studies were conducted to assess the properties of the estimators obtained. MSc student Akarsh worked on geographically weighted logistic regression based model calibration estimator of finite population proportion. He has obtained the estimators for finite population proportion in spatially non-stationary scenario. The developed estimators show asymptotic model unbiasedness as well as design unbiasedness. Spatial simulation study was conducted to assess the properties of the estimators. It was revealed that the proposed estimators are more efficient than the existing ones. Another MSc student worked on the estimation of crop yield at Gram Panchayat level using remote sensing and small area estimation techniques. Samuel has developed crop yield estimation method by applying small area estimation using auxiliary variables from remote sensing and GIS data. The obtained synthetic and composite estimators provide more efficient estimation of crop yield as compared to the traditional estimators. This approach may result in reduction of CCEs for Pradhan Mantri Fasal Bima Yojana. Another MSc student, Surya, worked on the development of 2 auxiliary calibration estimator of the population total. He has developed calibration estimator for population total using 2 auxiliary variables under 2-stage sampling, assuming non-variance function of the study variable. Simulation results demonstrate the efficiency of proposed estimator as compared to the existing ones in terms of percentage ARB and percentage RRMSE. This validates its efficacy as a reliable alternative for accurate population total estimation contributing to the field by enhancing precision through multiple auxiliary information. These are some of the conference presentations and trainings attended by our students along with one research paper. Third area is statistical genetics. Only one student, one PhD student worked in this area. Shubhrajit worked on statistical model-based prediction of discrete and continuous trait response in evolutionary distant species using genome-wide variants. So the major findings are identification of optimal combinations of variable selection and learning methods for predicting both discrete and continuous trait responses using simulated data, discovering genome-wide significant variants and their interactions to establish a genomic selection model for breeding value prediction, development of a GS model for predicting multi-trait mixture responses. The student has received the popular NAHEP CAS fellowship and visited Louisiana State University Laboratory as a research scholar. He has also received ICAR, JRF and SRF. Some very important publications came out of his study and published in very high impact factor journals. The last theme area is statistical modeling. Our PhD student Prabhat worked on singular spectrum analysis based on decomposition models for agricultural price forecasting. The objectives were to develop hybrid time delay neural network model based on SSA and EMD techniques and to develop multivariate SSA coupled with MGARCH model. The student has developed such models and all the models developed outperformed the existing comparable benchmark models. Another PhD student, Sandeep, worked on wavelet-based nonlinear time series models for capturing volatility in agriculture. He has presented his he has appeared for merit medal competition days for yesterday. His findings include wavelet-based hybrid time series models, accurate predictions regarding price trends and rainfall patterns using developed algorithms. There is a myth that using highest wavelet decomposition level will enhance the performance. Using his developed models and data, the student has proved it wrong, and he has also developed some R packages. Our MSc student Siddharth worked on XPAR and XPARMA models to capture cyclical nature of the data. He has done the necessary derivations and theoretical expressions were obtained. Out of sample forecasts were made using exponential autoregressive models, and the metrics indicated an impressive performance over ARIMA model whenever the data exhibited significant cycles. The R package developed by the student is well received by the researchers. Another MSc student, Shubhankar, worked on time series models for nonlinear intervention efforts. ARIMA-X, ANN-X, and incorporating growth models is the output. The intervention here was ban on blending in mustard, introduced in October 2020. Student has used his developed models and examined this intervention effect, and the developed models outperformed all the existing models. Our last MSc student, Shubhradeep, worked on modeling agricultural price volatility using Garchato models. The aim of the study was to develop an algorithm for finding jump volatility of Garchato model for modeling high frequency in agriculture. The findings is obviously superior performing Garchato model that makes use of discrete wavelet transform for estimating agricultural price volatility. These are some of the publications by the modeling group. These are the R packages. As you can see, the number of hits is quite high, though these are developed very recently. These are some of the conference presentations of the students. Now coming to placements, our students are well placed. 2 of them are placed at ISS. They have joined this year. All the 10 seats were occupied by our students in ARS, and they have joined. Assistant Professor 2, and 1 each as biometrician, rural agricultural extension officer, agriculture field officer, and assistant FCI. 1 has recently taken relief and joined the Central Silk Board, and 1 has very recently selected for RBI. As a follow-up, last year degree recipients, they have published some good papers. There are 7 of them, more than 6 NAS rating. These are the total publications by all our students, including the degree recipients. This is the distribution of those publications. And one student received Best Poster Presentation Award, and our students actively participated in All India Agricultural University Youth Festival as well as Sports and Games Meet, Inter-University Sports and Games Meet, and these are some of the certificates. Now we usually conduct informal alumni interaction with the ongoing students, and some of the topics are recognized, identified there, which will be given more importance in, especially for practical sessions later on. So online and offline we used to conduct with, especially with MNCs. And our students, especially the second year PhD students, take a lead in organizing many of the events in our institute. And Walkathon was organized on National Statistics Day, which came to IRA also. And National Science Day was organized, and International Women's Day was organized. These are some snaps from 2023 Convocation as well as orientation program of 2023. We try to bring out the hidden talents of the students using these competitions during the events. Poster competition, logo designing competition, sketching and painting competitions. The great statistician Professor Vikas Sinha visited our institute on a Sunday and given a lecture and blessed our students. We celebrate all other occasions as well. Our students participate actively in cultural programs as well as Rangoli making, etc. Our faculty is also not behind. They received some of the recognitions during the year. I am talking only about the PG faculty here. These are the research publications by at least one faculty member is involved. We are organizing several training programs and workshops as well. Thank you. I would like to thank Director and Dean ICAR, IRI, ISRI, PGS, PSC, VOS, Exam Committee, Seminar Association, faculty, degree recipients and students. Thank you all.
Thank you, ma'am. I think your presentation got maximum claps, ma'am. I do not know why. So I request Girish, for salient points of your studies, and we begin with that. So Girish has
a very good afternoon
15 minutes,
to each one of you, respected chair of the session, honorable director, esteemed dean, conveners, distinguished faculty, head of division, professors, and my dear students. I am here to present the significant educational achievements of 2023-24 in the discipline of bioinformatics. This course is a very new course in the graduate school of IRI. We started MSc Bioinformatics in 2011-12 and PhD Bioinformatics.
Dr. you take mic.
2014-15. Total faculty strength, number of PhD guide is 13 and number of MSc guide is 16. Total student strength, currently we have 39 students and PhD students enrolled are 32 and MSc students enrolled are 7. And so far, 13 students have passed out as a PhD, 14 students as MSc, and this year 9 students are going to get the degree in PhD and 6 students are going to get the degree of MSc. These are the list of the PhD students, 9 PhD students: Sneha, Saran Basappa, Soumya, Ritvika, Dipro, Nitesh, Vaibhav, Sweta and Anubhav. They are going to get the degree in this convocation. The first problems we have started in the area of theme of artificial intelligence, machine learning and statistical techniques in bioinformatics. The idea here was, the title is the development of computational approaches to understand the plant pathogen interactions. As we all know, the importance of the protein-protein interactions, and moreover there is a lack of prediction tools for studying these protein-protein interactions in the plant pathogen systems. Keeping that in view, the first objective was evolved to develop an empirical model to predict protein-protein interactions between plant and pathogen, and then this model was compared with an existing model for prediction, and this was applied in case of protein-protein interaction between wheat and pathogen, and finally a prediction server was developed. As we all know, what has been done is the data around 1400 pairs of plant pathogen interactions involving bacteria, virus, fungi. They were collected from the public available domain data like pathoplant, PHI base, SP, IDB 3.0, and those were scrutinized, and then after that, wherever there was a lack of sequential data, they were obtained on the basis of those identifications from the different protein databases, and different features were extracted on the basis of biochemical features, and then different machine learning techniques have been used, be it a random forest, be it a SBM, be it a artificial neural networks, and all kind of things. And then depending upon their predictions, the ensemble models have been developed, and then once this model has been developed, this model has been applied in case of wheat crop, as I said. And here what has been done is basically that the genomes were mapped after genomic data, the good after quality checks, good reads were used, and those were mapped, and transcript was obtained, and transcripts were even translated into the proteins, and they were used as an input to understand the interactions. So essentially, this web's prediction servers have been done. So as salient finding is that a model was developed to protein-protein interactions predictions, and a web-based applications or web resources have been developed to this. The second work was on computational intelligence in the discovery of natural products from agriculturally important metagenomic data. As there were a limited availability of the reference data, so metagenomic research has come in a big way. The first objective was to develop an approach for binning of metagenomic data using advanced learning tools, and then develop a framework of AI/ML technique for identifying the natural products in the bins of metagenomic data, and finally to identify natural product data in agriculturally as a part of an application. Here again we have in a metagenomic data. After getting the metagenomic data, a feature was extracted, and bins, they were binned and grouped into that, and then bins were evaluated. From these different bins, profiling had been done, and in that profiling genus species were collected, and then they were used as a from the reference database, and natural language processing techniques have been used for dimensional detections and all, and AI/ML techniques have been used to classify not all the natural products. There are only the 5 categories of the natural products were classified into 5 categories, and this has been applied on a real-time data on a kind of a rhizosphere, and this research was presented during the gold medal presentation. So essentially, findings are 2 deep learning based models were developed for a binning approach, and logistic regression based models or machine learning models were used for identification of natural products. So this tools, natural product predict tools, can be used for either of these 3 types of files, and from there one can predict these if the tools is confident that it will be able to predict the confidence with a more than 90%, otherwise it will say that none of these classes, and even the properties can be predicted. The third is the development of database for genes and gene family responsible in nutritional traits in field crops. As we all know now, everyone is looking for a kind of nutrient rich crops. In that case, like nutrient process like transportation, accumulation. So here the objective was collation and curation of genes and gene families responsible for nutritional traits in selected food crops, and a methodology was developed for classification of nutritional related diseases, and finally a interactive database had been developed. In this case, data were collected from the publicly available domain data, that is universal protein data. Well, features were extracted on kind of like amino acid decomposition, CTD, amino acid composition features were extracted, and then they were compared with various machine learning techniques, and one of the best technique has been used for classification purpose. And this is the database have been developed. So random forest unambiguously out of all the machine learning techniques have given a both binary as well as multi-class classification with more than 90% accuracy for both classification, and the NutriTrade database web resources is available to the public, and of course probably it is first of its kind to provide a comprehensive non-redundant catalog of nutritional trait related genes with supporting evidence from published literature. Next thesis is related to development of advanced learning tools for classification approach for fungal metagenomic data. This is restricted to the fungal metagenomic data because very few studies have been reported regarding development of deep learning based algorithms for using ITS barcode classification. Here the objective was to develop an efficient advanced learning based approach for molecular market and classification for fungi data from metagenomic data sets and empirically evaluate and develop a kind of a software. Here again a deep learning based funbar, CNN funbar has been developed, and they have mostly in a convolutional neural networks, and they have tried to, as usual, previously you have said the features, Kramer frequency features have been there, and model have been developed, and finally a web applications has been done. And here you can get a classification on the basis of species, genus, level kind of things. So first points indicates about the architectures kind of things, or that how many layers are there, how many filters are there, which activation functions is there. And then it also says that when you are going to get the highest classifications accuracy in the terms of that, and the CNN funbar on basis of the convolutional neural networks outperforms the competitive machine learning, be it a SVN or be it a KNN or naive Bayes or any random forest on as well as existing ITS classification softwares. Next is the development of advanced learning model for prediction of epigenetic modifications in crop. And recently much of the research are focused on the epigenetic kind of things, and most of the prediction tools are based on vertebrates. So there are very less information available about epigenetic changes in agriculture crops, especially in wheat. So here the student has tried to develop the advanced learning model for prediction of epigenetic changes in crop plants, and to develop an integrated model using developed several models have been developed, the ensemble approach for the prediction of epigenetic, and this developed model was used or applied to predict and study the epigenetic changes in the bred wheat, and finally a prediction tool based on the proposed model to predict epigenetic changes has been developed. So here advanced learning models were developed, and as far as feature extraction is concerned, so AMIP, that is average mutual information profile, along with dinucleotide frequency and other features were, or say guanine cytosine content kind of things. Several best performing models, be it a support vector machine, random forest or gradient boost, were then integrated using ensemble learning approach. So here after the sequence data we have got a kind of a feature extraction, then we have selected the features, we combined the features from different techniques, and the combined or you can say intersection of the features were used for training and testing, and then we have developed a model, and that model can use for a kind. Apart from the two prediction servers, two R packages were also developed under this particular paper. And after analyzing the promoter region, as a part of applications, the LBD domain containing genes, 6 species were found, and found that the 6 methylations was higher in the Tritiaceae family than in the rice and Arabidopsis. And another findings, it is saying that within Tritiaceae family, it was observed that with the evolution, the rate of methylation has decreased, and that's why the highest is in the monoploid and the lowest is in the hexaploid. Next is the kind of a deep learning algorithm for identification of copy number variations, lack of well-established pipeline for data analysis and improved algorithms, high FDR due to non-uniform results in CNV. So the objective was to develop a deep learning based model for identification of putative copy number variations, and then evaluate the performance of these developed models, as well as to develop a web-based resources or pipeline for the developed methodology. So here the study has been done on the equipoints, and the blood sample from 6 equipoint breeds have been taken, and sample were genotyped with the exomes equine genotyping array, and the developed methodology is compared with the existing machine learning techniques like SBM, random forest, linear regression, naive Bayes, and all kind of things. Ensemble methods were shown. Essentially, this work is also the knowledge discovery as well as the product discovery, a deep learning based methods for prediction of copy number variation using SNP arrays has been developed, and the two web resources, CNP Atlas and prediction server, which can be a kind of a research accelerating tools assisting in improved traits of this, and this is also a kind of a first of its kind. The next study is based on machine learning based approach for long run non-coding RNA, subcellular localization predictions. Here basically the objective is to develop a AI/ML based approach for predicting the subcellular location of long non-coding RNA in view of the importance of long non-coding RNA, and then to implement the developed approach on a web server. So here they have obtained the data. After pre-processing of the data, they have tried to have a seven component of the cell. So data set of all the 7 components, which distributions of those 7 data sets in the different 7 subcellular component is given in the right side of that. And then they try to have a feature extraction, and the novelty is that they try to use the traditional features like sequence feature, structure features or physiochemical features. They try to combine it, and then with or without a kind of synthetic minority oversampling techniques with, and as well as the main novelty is that they had a motif features with a mismatch, and finally they developed a model. And once they developed a model for this long non-coding RNA, they have used for the black paper. And here, if you look at the data set, so random forest appears to be, there are a series of like almost 9 models, machine learning models have been used, and among that, random forest appears to be the best model. So, first thing, they developed a new feature extraction method based on motif frequency, which I mentioned, with mismatch allowed using local alignment dynamic programming. Then secondly, they developed a random forest multi-class classifier to predict this long non-coding RNA subcellular localization. As I said, only 7 classes with 93% accuracy, and they have developed the web servers for the prediction of subcellular localization, as well as they have also developed a web resources which is of its own kind. Then the last study of PhD is the comparative genomic, last but one is a comparative genomic study for domestication related traits in beginner species. This basically we all know beginner species have numerous benefits, but there are certain undesirable domestic traits such as pod shatterings and photoperiod sensitivity. In view of that, the first objective was to identify the genes and the gene family involved in scattering and photoperiod response traits in beginner species, and then to develop a system for genome-wide identification of cis-regulatory elements and enrichment in beginner crop, and to carry out comparative genomic analysis of domestication-related genes and gene families in beginner species. So here the candidate orthologs genes and gene families involved in scattering and photoperiod response traits in model and non-model species were first identified in beginner species based on the comparative analysis and syntax study, and also cis-regulatory elements enrichment analysis has been done to understand the conservations of cis-regulatory element across crop plants, and these are the 2 databases. So the first thing we try to they try to identify the potential target loci to overcome the scattering as well as photo sensitivity traits in the beginner species, and this Synteny network analysis highlighted the evolutionary conservations of scattering or non-scattering genes across species including beginner, and it also shows that the strong conservation of photosensitive and photoinsensitive groups of genes in Synteny among orthologs. And it has been, as I said, that all the data generated from the present study had been integrated, and a database has been developed which contains detailed information about genes related to scattering, non-scattering and photoperiod response. It has been proposed to apply the genome editing tools on the identified target genes in 4 beginner crops to have further insights into their role in non-scattering and photosensitive phenotype, thereby accelerating the domestication. Last PhD study is related to development of genome-phenome browser for rice cultivars. Here, the idea was to collect and mine data on genes involved in different trait expressions of rice, and to develop a tool for integration of genome-phenome data and develop a genome-phenome browser for identifying traits of rice. So, in this study, of course, relational database has been designed for the information obtained in objective 1, and the major findings is that information organized can be used to enable direct comparison with QTL information available in any database, which will facilitate a candidate gene approach to identifying the gene responsible for QTL. There are 6 bright MSc students who are going to get this degree in this year. The first students is talking about the discovery of molecular marker and development of database for rice bean. Here around 5 lakh transcripts in rice bean seeds and leaf transcripts, as some of the data were generated and some of them from public domain data, and they have mined the SSR marker, 61,000 SSR marker, close to 2 lakhs SNP markers were discovered, and out of SRR marker, they are saying that dinucleotide repeats were predominant around 3/4. So molecular marker database for IceBeam and a web-based software to access the database has been developed. In the second study, the tools for selective SUPS analysis has been used using artificial intelligence, and the tools uses the can accept the VCF file format, chromosome number and region of interest in chromosome, and on the basis of the input and output can be provided either no selective SUP or selective soft selective or hard selective. Here the web tools known as ICAR CRISPR has been developed for off-target prediction influenza. The output is that web tool can be used to predict whether a given target DNA sequence is on or off target for a given sgRNA for crop plants, and the models for these tools have been developed in the earlier thesis. And a semi-supervised approach for binning for metagenomic data. Here again, the developed semi-supervised approach performed with more number of features, mean of AMIP, log transform guanine cytosine contents, HNF, etc. And the research outcome is that the developed approach uses this autoencoder to extract the relevant features and as well as for dimensionality reduction for binning. And the next is on the similar line on the novel and efficient pipeline for metagenomic binning. Here the output is that the developed approach can make clusters with many contigs, whereas the existing softwares where like they are not able to do more than 1500 contigs or less than 1500. So those problems are not there, and moreover this is more performance is also better. And development of an approach for the identification of core microbiome being an important. The important thing is that they have developed a core microbiome R package that presents a significant advancement in the field of microbial community analysis because it integrates diversity analysis and significance testing. As far as publications is concerned, there are 71 publications, 5 training manuals, 5 manual books, and most of the 24 publications are or you can say that more than, between 10 to 12. As far as student publications are concerned, out of this, only 11 are student publications. They are listed here. As far as students have got a different award in different and faculty have also one of the faculty has got Young Scientist Award. And we have conducted 3 training programs, and almost 130 students were trainees have undergone these training programs, 2 CAP training programs and 2 training programs on CRP genomics. From a core of the heart, I would like to thank the Director and Vice Chancellor ICAR-IRI, Director ICAR-ICRI, Dean, graduate schools, faculty members, TAC, and one and all. Thank you very much.
Thank you, Girish. Thank you. We'll have, towards the end, we may have a little bit of interaction with the professors. Now, Alka ma'am.
Dignitaries on the dais, of the dais, my dear faculty members and students, good afternoon to all of you. I am here to present the significant educational achievement in the discipline of computer application at ISRI. Coming to the faculty and the student strength, we have 13 research guides and 12 guides for the PhD. Majority of the faculty members are placed at ISRI, but some of the faculty members are in other institutions also, like NIAP, and we are faculty members at IRI also. In this convocation, 8 of our students are going to get the degree, and then the major themes in which they have worked is artificial intelligence, machine learning, deep learning, and its applications in agriculture, DSS, and the mobile-based applications in agriculture. This is the list of the MSc degree recipients. 5 of the MSc degree students are there. Now coming to the work of the MSc students, the first student, Sakshi Rawat, she has presented for the IRI Merit Medal also. She has worked under the guidance of Dr. K.K. Chaturvedi on the topic development of web-based bibliometric analysis tool for the agriculture. So in this particular case, they have developed the novel bibliometric indicator named as author performance indicator. Based on this tool has been developed based on the which quantitatively evaluates the ICAR scientist performance based on the research paper. They have taken primarily data for this particular thing from the Krishi knowledge repository. So they have given this tool as a name Krishi Bar, and this tool is available at this particular URL. Using this particular tool, different types of analysis can be done, and these are some of the screenshots of this particular tool developed. Coming to the second student, Rohit Vanshraj, he is done under the guidance of Dr. Shashi Dahiya. He has worked on development of DSS for energy budgeting of the crop production. So they have come out with the mobile-based DSS, that the aim for this particular DSS for the optimization of crop production inputs and resources, and for this particular thing they are utilizing various energy parameters, and then they are going for the energy budgeting in the crop production. This DSS basically identifies the different cropping systems that are suitable, sustainable, and as well as the reduce the ecological footprint, and it gives a different does the comparison of the different cropping patterns and present it in a visualization method. Shashi Kumaran S, under the guidance of Dr. Somen Paul, has worked on the development of web application for feature selection and crop yield prediction of landscape diagnostic survey data using machine learning technique. This landscape diagnostic survey data has been taken under the CISA project for rice and wheat crop in Bihar and eastern UP. They have tried with different feature selection methods for the like stepwise regression, lasso, and other methods. After the feature selection, they have gone for the model building using the machine learning techniques, and they have tried with the different machine learning techniques of multiple linear regression, support vector regression, random forest regression, and ridge regression. And with the analysis, they have come out that the random forest regression came out as the most accurate one for the feature selection, and for the crop yield prediction also, only with a couple of exceptions, the ridge regression and the stepwise regression has worked well for these particular methods. Bhaveshri, under the guidance of Dr. K.K. Chaturvedi, she has worked on the creation of a mobile application. This app has been developed. The content of this app are available in both Hindi and English language, and this app provides information about the beneficial insect species. This covers their details as well as the specific crops for which they are relevant and the proper geographical regions where they are available, along with the other information and the mechanism to handle it. Gaurav Mitra, under the guidance of Dr. Mohammad Samir Farooqi, he has worked on development of mobile application for pest surveillance and management of tomato crop. This work has been carried out with the data or with the collaboration with the NCIPM. So they have developed one mobile application with the name Pest Serve Tomato, and this particular app contains information about the common tomato pest, at the same time disease which are prevalent in tomato. This particular app determines the pest incident level, which are based on the number and severity of the pest present in the plant. So now coming to the PhD students, 3 of the PhD students are going to get degree in this particular convocation. Sapna Nigam, under the guidance of Dr. Rajni Jain, she has worked in the area of artificial intelligence, deep learning, and she has developed development of deep learning model for identification of major wheat diseases. Data for this particular study has been taken up from the IRI wheat program. They have collected more than 6,000 images for the healthy as well as from the diseased plants. The thing is, they have come up with they have proposed a fine-tuned efficient deep learning model which achieved the accuracy of more than 99%. At the same time, they have attempted with the severity estimation also with the disease, within the disease. So this severity estimation model also achieved the testing accuracy of more than 96%. And then the best part is that all these deep learning models are integrated in this mobile application with the name AI Disk, Artificial Intelligence Based Disease Identification System for Crop. This mobile app is already released from the ICAR, and this is in use in the real-time diagnosis at the farmer field. Now coming to the next work of Lakshmi Sonkusle, she has worked under the guidance of Dr. K.K. Chaturvedi. She has worked in the area of text mining and worked on study of text mining approaches for topic discovery in agriculture. Particularly for the agricultural data set, they have selected 3 different types of data sets. COVID-19 and agriculture, crop science and animal science domain they have selected, and for this they have collected publication material from the Google Scholar, at the same time from the Krishi repository also. They have tried up with the different combinations of the pre-processing methods and different text representation techniques also they have tried. Then they have come up with the different models, LDA models they have tried, and compare it with the existing methods. This novel approach which they have proposed in all the 3 cases for the agricultural data set has given a very good promising result with more than 70% accuracy. They have developed or they have integrated this particular software into this dashboard, which is available on the web. Coming to the next student, Murari Kumar, he has worked on natural language processing based interactive system for IPM in rice and wheat. He has worked under the guidance of Dr. K.K. Chaturvedi. They have collected for this particular study, they have collected the data from the Kisan call center. So because it is a very huge data set, so they have created a web scraping tool. Using this web scraping tool, they have fetched the data and then they have come up with the name entity recognition model for doing the text annotation part, mainly for the IPM data. And this model has received the accuracy of more than 97% for the same. With this, after that, they have gone for the intent classification. For the intent classification part, they have used the ensemble of the machine learning models. At the same time, they have tried up with the deep learning models also. And coming to the results part, for the intent classification of queries into the IPM and non-IPM, they got the accuracy of more than 95%. Similarly, for the deep learning algorithm also, by LSTM performed higher over the LSTM, and they have integrated this into a web-based system. These are some of the student publications. Now coming to some of our students got the overseas exposure. The Himanshu Shekhar Chaurasia, he is currently doing the PhD. His paper was selected and he has gone for the oral presentation in the 6th International Rice Congress at Manila, Philippines. He got the opportunity to go for the under NAHEP caste component. He got the opportunity along with Mohit to go to the University of South Australia, and they have spent almost 15 days there. Similarly, Preeti also got the opportunity to visit the University of Maryland, Baltimore under NAHEP caste project. Coming to the student placement, one student is already in the scientific services, 2 are working as an RA and SRF, and 4 students are currently pursuing the PhD program. Coming to the publication from the discipline, we have got a good publication, 22 publication under different dance codes, along with the technical resources. Now, our faculty members are involved in the ICAR annual day. Some of the technologies were awarded. In this, 3 of our technologies were selected, KVK Portal, AI Disk and the KCC Chakshu. And for 2 of the technology, AI Disk and KCC Chakshu, it was selected to be given the award from the Honorable Minister, sir. So some of the faculty member has got the best oral presentation awards also. Dr. Sudeep, Dr. Chandan, Dr. Sabbar. Myself and Dr. Sumit got the opportunity to attend one workshop on the digital survey tools in Thailand. Now, sir, I would like to mention here some of the very significant digital initiatives in which are particularly in the agriculture education. They are undertaken in the NAHEP Component 2 project at ISRI, and almost all our faculty members are involved into it. And because these are of the academic interest, like this academic management system which is already in use at IRI, this has been implemented in more than 60 other universities also of the NARS. We have come up with the e-learning portal. So this portal has gone for the content creation. It has got all the textual material which has gone for the peer review process. In this particular case, 77 PG and 165 UG courses are available, and this is becoming very popular, and more than 90,000 downloads are already there in this particular URL. So other faculty members and students can be benefited out of it. Similarly, under this NAHEP Component 2 project, a lot of strengthening has been done in the state agriculture universities. So in this, virtual classroom has been set up in almost all the state agriculture universities, but at the same time at ISRI, we have created an Agri Diksha channel also. So all the lectures, video lectures which are recorded in this virtual classroom, they are available in this Agri Diksha channel, and the students and faculty can be benefited with the lectures from the other faculty from the other universities also. At the same time, sir, under this project, AR/VR experience centers has been created. So we know this immersive learning is now becoming very important, and this is the first time this has been attempted in the agriculture education. So in this particular thing, already 14 VR modules are developed, and this is available on this particular URL. Another significant achievement which has come up is the blended learning platform. So this blended learning platform, which is an integrated platform which provides opportunity to go for the blended teaching methods. So this has been implemented in all the state agriculture universities. At the same time, lot of capacity building is already there in this particular thing. Along with this, one major initiative, that is creation of the other core content, which we call it as containing the audio-visual component, 2D lectures, video lectures, animation lectures. So these are being created with the help of the external agency under this particular project. So we are planning to create some standard 60 courses which are part of the BSMA, and out of this 16 are already available. This has been hosted in the BSMA, like our BLP platform. These are some of the snapshots of the VR modules which has been developed. This is the listing. These modules are developed in various disciplines. Some of the glimpses of the same. Sir, our faculty member was engaged in organizing some of the workshop and the conference, and the significant one to mention is we have organized one international conference on blended learning ecosystem for the higher education in agriculture, ICBLE 2023 in March. Under the NAIP Component 2 project only, and in which this BLP platform was released by the Honorable Minister Sir, and a lot of international and national delegates participated in this particular workshop conference. Similarly, like we were talking the BLP, a lot of capacity building has undergone for this particular part, and we can say more than 7,000 participants, including students and faculty members, has been trained for this particular thing. Other than this, sir, we have organized other training programs also. And then using this blended learning platform itself, we have organized some of the training programs like in a both line mode, like hybrid mode. We have gone for conduct of this training program. Some of our own program, at the same time, some of the program we have organized. For example, one program we have organized in collaboration with BITS Pilani. We have organized training programs under HRM, at the same time CAP training programs. And at the same time, sir, this time we have organized one training program on data science in agriculture. So this we have organized on our own initiative, and this was well appreciated. Our students are actively participating in many of the activities which we are organizing. And like my fellow professor has also mentioned, we are organizing the interactive webinars with the different alumni. So in this series, starting from January, we have taken up the initiative to have one alumni talk every month. So we had one in January from our proud alumni Ashutosh Karna, who is working as a principal technologist at HPL Corporation in Spain. So all those mentioned here are acknowledged. Thank you so much. And coming to this code, because our division and mainly ISRI is doing a lot of work in the area of artificial intelligence, which is going with the thought of our Honorable Prime Minister also. And we are collaborating in many, partnering with many organizations like IIT Ropar, PAU Ludhiana and NABARD. Similarly, we are working with POCRA also to take our initiatives which has been already developed to take it to the farmers field, at the same time to come up with the new initiatives. Thank you so much.
Thank you, ma'am, and thank you for your wonderful presentation. Now, Monica.
Thank you to all the professors. I now invite respected chairperson for his kind remarks.
For 10 minutes.
Dear students, please be seated for 5 more minutes. Students, please be seated.
Honorable Director of this great institute, Dean Madam, Monica ji, Sriti ji, Joint Director Dr. Padaria Sahab, senior professors, faculty and dear students. At the outset, let me very profusely compliment the professors who have presented their significant findings. Very beautifully presented format. Time was a constraint, but you have been able to make your point. And the salient observations from each presentation I have been just captured. Maybe we will try to share with you. Friends, just for your kind information, Director Sir had mentioned that 1973 I joined this institute, and when I come to this platform, particularly this dais and this auditorium, it takes me back to 50 years. In 1973, when I joined IRI, the first event was introduction of students, and on this platform, everybody was called, and then Dr. Dadlani sir conducted that event, and each student was asked to do something, and I was asked to do. Sadamate, you have joined IRI, but when you were in Maharashtra, in your village, in your tehsil, what sort of activities you were involved in, number one, and number two, what prompted you to come to IRI? Okay. The second thing was very interesting, because when I was studying in Pune Agriculture College, Dr. Joshi was the director of the institute, and my professor was Dr. Thorat. I never thought that I will join IRI, but Dr. Thorat said, Sadamate, you have a lot of interest in extension activities, and then you just send your application, and then we will try to see. So some seniors were here. I came to IRI, but the fantastic part of IRI learning is the very sound interaction between the juniors and seniors and the faculty. And that, gave me a very good platform to grow in my own field. And as Sir had mentioned, that I had a chance to work with Krishi Bhavan, with ICAR in the beginning. 1978 batch of ARS, I believe it's Dr. Ayyappan's batch, if I remember correctly. And then Krishi Bhavan, Planning Commission, some national, international assignments. So that has given a good platform. And my young friends, you're very lucky to be in IRI, get a brand of IRI, get a degree from IRI. I mean, it's your luck, and it is your fortune in future. I mean, having said that, let me come back to some salient points from each presentation. Fortunately, I know the professors and the faculty very well. By being around Delhi, around IRI for the last 50 years, I make it a point to attend important events of IRI. Dr. Padaria sir will agree with me that I believe I am the only one who must have attended all last 50 Krishi Melas, if I remember correctly. And so also, I make it a point to attend, your convocation, your events, and sir and madam had given me a wonderful opportunity of being on the, academic council, then, adjunct faculty, etc. Now let's come to, the presentations. I must compliment all the presenters. As I mentioned in the beginning, they were very concise, very focused, and you have been able to manage within allotted time. Pramod ji has given a very salient points on climate change and economics, management of pond technology, then economics of DSR, micro irrigation, and so on and so forth. But this economic analysis of new technologies is far more important, because when you move from laboratory to the field, the dimensions change, the parameters change. You get more towards the resources, more towards socioeconomic factors, so on and so forth. And that is the, strength of, technology. If the field is bright, the lab gets, better signal. And, that is what, Pramodji has, pointed out very clearly. He mentioned about, ENAM. And, also mentioned that, ENAM application has been increased, 17 times. I mean, that shows the, strength of the technology, number one. Number two, acceptance, by the Indian farmers, whether he is small, poor, medium, or, resource endowed. 17 times, ENAM application has increased. And, one more, very interesting point which, Pramodji mentioned is that, I mean, how these FPOs are coming up. Empowerment of farmers, farmers getting on aggregated mode of production, aggregated mode of marketing, intervening on aggregated FPO lines, cooperative lines, farmer group lines, farmer commodity organizations, so on and so forth. And having been, with the Ministry of Agriculture, with Planning Commission, couple of international organizations, I mean, I have seen, particularly in, Europe, in Latin America, in US, Canada, etc., these commodity organizations and commodity groups, will be the, future drivers of, technology dissemination. Let's come to the ninth, sir. My own subject, very interesting subject, and I'm in touch with, Nain sir, Dr. Padaria ji, very closely as adjunct faculty, and, a good number of times as examiner of the, MSc students, PhD students, etc. A couple of points which Nain sir has mentioned is, entrepreneurship development and how it is taking a shape and size in agricultural extension services. Then, entrepreneurship is being promoted at the highest level. Honorable Prime Minister is emphasizing time and again the importance of, startups, entrepreneurships, public-private partnerships, so on and so forth. And these, the parameters are very important when you talk about, agriculture, research, technology, dissemination, farming population, so on and so forth. Then, Dr. Nain also mentioned about, social media application. Also mentioned about, Nutri-Smart Village. I mean, here I'll take a minute. All our KVKs and, field extension services are promoting, home-scale nutritional, the challenges. I mean, that is, very important because, we are self-sufficient in food and food production. Stocks are strong enough, but we may have to see, and as a young bright minds, from IRI, I mean, see how the technology has percolated down to the, weaker sections. Perhaps they need a different kind of, social and economic security. And when it comes to economic security, food security, IRI technologies and IRI science, IRI application has shown very clearly that even at that level, IRI makes a difference. Then, let's come to, Sini Madam's presentation. Madam, you have, very clearly pointed out the different types of designs, their efficiency, then, the probabilities and how they could be used, analyzed, for the, crop estimation, for the, climate change variations, so on and so forth. Girish has, highlighted on AI and ML, artificial, intelligence and machine technology. I mean, this is, these are the areas which will lead science in a future perspective. You also, mentioned about nutritional trials and how AI and ML can be applied. Then epigenetic changes and how AI and ML can capture it. Lastly, Alkaji, I must compliment you. You did very well. And 3-4 areas you mentioned which appeal to me and I may share with you that, computer application in, decision support system. When it comes to, again, being student of extension, when it comes to, farmers and farming situations at different levels, you have, farmers with, very small holdings, medium holdings, resource poor categories, export-oriented type of farmers, and they need, different kind of, combinations and matrices, for decision support systems in their own, venture. You also, mentioned that how these web applications are, coming up and how they could be applied when it comes to dissemination of technology. Then, digital education, and I like, deep learning models. Having said that, and having, gone through, this, different salient observations, sir, I'll take, just, 5 more minutes if you kindly allow me. Is it okay? Yeah, there are, 4 or 5 points which, come out of this presentation, and our young minds must capture it. Number one is, how research and development is linked and where science can make a difference. Point number 2, how science can make a difference, how technology can make a difference to different social dimensions. I mentioned to you, there are different categories of farmers, different categories of, resource endowments, poor, rich, and how science should, intervene and integrate. Then third is, next generation agriculture. And our, this young mind, particularly, MSc, PhD fellows, students, they must appreciate the science is moving, very fast. And agriculture, should also travel fast. And application of science on the parameters which, professors have demonstrated, we should be able to capture and build the capacities of the, development departments, because, that is where, it matters most. Agricultural universities and institutions like IRI, can demonstrate what science can do. But, if it does better when the development departments take it down. Definitely, you are ahead of application for 10 years, but the development departments are, let's say 5 or 10 years behind of, science and application. And therefore, we all should be responsible, for developing capacities of the development departments, field functionaries across the system, be it is agriculture, horticulture, animal husbandry, dairy, fishery, poultry, because agriculture in planning commission's definition is, all land and water-based activities. Then media and ICT is another area. Then interplay between different systems, and lastly, the advanced applications like the drone, AI, ML. But what matters is how we build the capacity of the operators. There was mention about entrepreneurs, input support service providers, so on and so forth. And I, being with around agriculture system, for a pretty long time, I find that, there are different streams, scientific streams, development departments, input support providers, entrepreneurs, social, the mobilizers, non-government organizations, each has a place, to make agriculture move fast and ahead. I must thank, Honorable Director, Sir, Madam, Monica ji, Shruti ji for giving me this opportunity, and giving my small little thanks to you. I shall sir, wait for the some more opportunity for giving a little elaborating interface with you all. Thank you very much. Thanks a lot.
Thank you, sir. I now request Director, sir, for his kind remarks.
So mine was not on the schedule, but I made a request that I should be allowed to speak for a minute or so. I must say I thoroughly enjoyed the presentation from social sciences. And I would like to compliment particularly the three professors from ISRI, Srini, Alka, and Girish, led by their director, Dr. Ajent Prasad, who is present here. And also thanks to Dr. Prasad for bringing a good number with him. I was remembering a poem which we used to read, चल पड़े जिधर दो डगमग में, चल पड़े कोटि पग उसी ओर. This was said about Mahatma Gandhi. So he came here, he brought a lot of, the students with him. And I will request Dr. Prasad to come tomorrow also for the LBS lecture. Because you remember when we were students, for LBS lecture, this hall would be insufficient to accommodate people. All the side steps will be full, corridors will be full, and these both sides, it will be completely full, so much so that even people will be sitting here in front. There have been lectures which we recall. I want to see that kind of response tomorrow. And then we used to take this opportunity to say that IRI needs a bigger auditorium. Okay? So tomorrow, since our Director General is going to speak, we will take this opportunity to make that request before him as well, if we are able to manage the numbers so well. But leaving that aside on lighter note, the presentation which was made by three professors from ISRI, I must say that there is so much of capacity available within our system that we don't need to look far. Actually, I must say here that all our scientists who are working in biological sciences, working on QTL mapping, genomics, gene sequencing, genome sequencing, etc., must have a very close collaboration, NVPGR including, must have a very close collaboration with bioinformatics group, with a statistics group, and computer application group. This is essentially required, which is at present is not really happening. Many times we have so much of data, genome sequence data available, 3, 4, 5 sequences of different varieties. You talk to people, the data is already available. How to analyze, how to make sense out of that, then we look beyond our institution without realizing that we have so much of capacity here. When Girish was making presentation, he is economist. Economist talking about, genomics, metagenomics, sequence information. If you close your eye for a moment, you will not realize that economist is talking to you. So that can only come with a deep involvement, because he has recently joined there as head of bioinformatics, and within that time his domain was very different when he was in IRI, but understanding this whole gamut of bioinformatics and bringing it in the right perspective, presenting it as if he is deeply involved into subject, and he knows so much of biology about genes, genome sequence, and so on and so forth, which is very hard to really believe. So this is really something very encouraging, and I compliment each one of you, and we look forward for very close ties and collaboration in this field. That is essentially required. Thank you so much.
Thank you, sir. I now request Director Sir and Dean Madam to kindly felicitate the chairperson, to be followed by a group photograph of the professors of this discipline, please, on the stage.
So with this, we come to the end of the 6th session of School of Social Science. I would like to thank the chairperson, Dr. Sadamate, please. Thank you, sir, for giving your insights. Students, please, 2 minutes. खत्म करने दो, बैठ जाओ. So this was a marathon run for us. For 2 days, we were involved in presentations by different professors of the 6 schools. I would like to thank all the chairpersons who chaired the sessions and all the professors of 26 disciplines who gave the what students are doing, what are their research achievements. Thanks to students also. Because of you, today it's a vibrant atmosphere here. Thank you so much. I would like to thank the students, volunteers from Seed Science and Technology. I'd like to take their names: Ambu, Samrat, Sushma, Sushmita, Shreya, Ayushi, and from Food Science and PhD, Prasoon, Brijesh, Rajan, Sashil, Lakshmi and Sindhu, and Mr. Dharampal, Senior Technical Officer from Seed Science and Technology, Mr. Ramesh and Ankit. Thank you all. Thank you so much.
Dear students, please reassemble at 2:20 because we have lecture at 2:30. So all kindly grace the occasion. Thank you.
I deem it privilege to request the meritorious awardee to kindly join the dignitaries on the dais. Esteemed Chair of this session, Dr. S.K. Chaudhary, Deputy Director General, Natural Resource Management, ICAR, Honorable Director and Vice Chancellor, IRI, Dr. A.K. Singh, Meritorious Awardee and Speaker of the session, Dr. Y.S. Chive, Principal Scientist Agronomy, Joint Director Education and Dean IRI, Dr. Anupama Singh, Joint Director Research, Joint Director Extension, Project Director WTC, distinguished guests, school coordinators, heads of different divisions, professors, research scholars, students, ladies and gentlemen. A very good afternoon to each one of you. On behalf of the Graduate School IRI, I am delighted to welcome you all to the 23rd Sukumar Basu Memorial Award Lecture. This award commemorates the memory of late Sukumar Basu, who was for many years concerned with various aspects of agriculture and also associated with ICAR in the capacity of Secretary, Chairman, and Vice Chairman. This prestigious award recognizes outstanding research contributions in agricultural sciences, including animal husbandry and forestry. I now request Honorable Director IRI to kindly welcome the esteemed chair of the session with a bouquet of flowers. I now request Dean Ma'am to kindly welcome the speaker of this session, Dr. Y.S. Chive, with a bouquet of flowers. I now request Dean Ma'am to kindly do the honor of presenting the formal welcome address. This is not possible.
Thank you, Dr. Rao. A very warm welcome to one and all to this auspicious occasion of the 23rd Sukumar Basu Memorial Lecture Award session, of which, for which the speaker of today is the recipient of the award, our very beloved faculty, Dr. Y.S. Chive, who was competitively selected based on the applications which were received and which were judged by a very high power committee constituted by the Honorable Director IRI. And on this occasion, I am particularly thankful and I welcome the DDG NRM, esteemed chair of session today, Dr. S.K. Chaudhary sir. Sir, we welcome you all. On behalf of IRI, kindly accept our greetings. So, about the Sukumar Basu Memorial Award, although Dr. Rauf has made my task little easy because he has explained a little which was there for me to explain. Nevertheless, this Sukumar Basu Memorial Award has been instituted for outstanding research in the field of agriculture, including animal husbandry and forestry, that lead to increase in the agricultural productivity. This is a biennial award which carries a cash prize of Rs 25,000 and a commendation certificate to be met out from the interest which is accrued on the amount donated by late Dr. Sibdas Basu to commemorate the name of his brother, late Dr. Sukumar Basu, an Indian Civil Services officer who for many years was related with the various aspects of agricultural research and education at the country level, and was also associated with ICAR in particular in his capacity as its secretary, vice chairman and the chairman. Now, not taking much time, I request the Honorable Director IRI to kindly introduce chair of today in brief with the audience. Thank you, sir.
Very good afternoon, distinguished chair of this prestigious Sukumar Basu Memorial Lecture Award, Dr. S.K. Chaudhary, the awardee, Dr. Sive, my colleague, Dr. Anupma, and in the audience we have with us Professor A.K. Singh, former Vice Chancellor, Rajmata Vijay Rajesh Scindia, Krishi Vishwavidyalaya, Gwalior, and Professor Jalali, former Director Research, Haryana Agriculture University, Hisar, and distinguished plant pathologist. Both of them are members of our academic council. We welcome you, sir, for this particular award lecture. All the students, staff, professors, heads of the divisions, students, it's my pleasure to introduce the chair to this distinguished audience. Dr. Suresh Kumar Chaudhary, as his full name speaks, we know of course as Dr. S.K. Chaudhary, is currently Deputy Director General, Natural Resource Management at ICAR, and he acquired his bachelor and master's degree from JNKVV, Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur, and doctorate from MPKV Rahuri, and he did a postdoctoral fellowship in the United States on Soil Salinity Laboratory, Riverside, California, Davis, and from the Institute of Soil Science and Plant Nutrition, University of Rostock, Germany. So very distinguished career. He joined as Assistant Director General, Soil and Water Management at ICAR in 2020, and as Deputy Director General after that. He also served at the Central Soil Salinity Research Institute in various capacities, and his core area of research has been soil physics, agriculture water management, management of salt-affected soil and poor quality water, climate change, impact on agriculture. Dr. Chaudhary is recipient of prestigious TOAS Medal of United Nations, Commendation Medal of IAEA, ICAR Rafi Ahmad Kidwai Award, ICAR Team Research Award, NAS Recognition Award, and Lifetime Achievement Award of the Institute of Engineers, 12th International Congress Commemoration Award of Indian Society of Soil Science, and many other societies award. He is having fellowship of the distinguished science academies, and including the National Academy of Agriculture Sciences and the most prestigious Indian Academy of Sciences, Bangalore. We are indeed grateful to you, sir, for kindly agreeing to be with us here to chair this prestigious award lecture, and thank you once again for accepting our invitation. Thank you so much.
Thank you, sir. I now humbly request the Honorable Chairman of today's session, Dr. S.K. Chaudhary, to kindly present his opening remarks and to introduce the meritorious awardee of this prestigious award to the august gathering.
Thank you very much. Namaste to all of you. Dr. A.K. Singh, Director of Indian Agricultural Research Institute, Dr. Anupama, Dean of IRI. Today's awardee, Dr. Shive. Today we have Dr. A.K. Singh, former DDG NRM and former Vice Chancellor of Gwalior University. Also Professor Jalali, colleagues from IRI, student friends. It is indeed a great pleasure to be here. Sukumar Basu Memorial Lecture is very, very important lecture of IRI, and it's also part of IRI Convocation. And I feel it is a great privilege and it's a great honor to me to chair this important lecture today. Today's awardee, Dr. Shive, is very well known agronomist who worked with Dr. Rajendra Prasad, and he has contributed a lot, mostly in the areas of nutrient management and the recent advances in agronomy he tried to apply it throughout his research career. Just briefly introducing Dr. Shive. He is PG from CS Agriculture University of Kanpur, then PhD from GBPUT Pantnagar, and he had excellent academic records all through. Then he went to China in Zhejiang, well-known Hangzhou University of China, and he did his postdoctoral there. And after that, he worked in the capacity of scientist, senior scientist, principal scientist, and also professor in Division of Agronomy, and right here in IRI. Dr. Shive is a fellow of NAS. He is a recipient of Best Teacher Award, both of ICAR as well as IRI, and also he is involved in several professional societies, including Indian Society of Agronomy, and he worked in various capacities and is recipient of various awards, including the awards from Fertilizer Association of India. Coming to the research part, he mostly worked in two different aspects. First aspect, the release of nutrients, particularly the nitrogen. So as I said, he worked with Dr. Rajendra Prasad. So without mentioning that he worked in neem-coated urea, I think it is not required. Since he was working with Dr. Rajendra Prasad, he worked in neem-coated urea, subsequently in sulfur-coated urea as well. And so far zinc nutrition is concerned, he was working in agronomic biofortification of zinc, and he has developed several technologies. And also in his career, he did a lot of experiments, a lot of students, and a lot of academic work, teaching and all. And he tried to develop some good understanding in the area of nutrient management, particularly the nitrogen and sulfur. Dr. Shive, I had an opportunity to see your biodata several times as an applicant of many awards. So I know many things. That's why without reading from here, I was able to summarize what you did all through your career. So without taking much time for the initial formality, I now directly request Dr. Shive for his presentation. Thank you so much.
Most Honorable Chairman of this session, Dr. S.K. Chaudhary, DGG NRM, Honorable Director and Vice Chancellor of this prestigious institute, IRI, Dr. A.K. Singh ji, Honorable Dean and Joint Director Education, Dr. Anupama Singh ji, galaxy of the scientists sitting off the dais,
project directors, head of the divisions, professors, scientists, ladies and gentlemen. At the outset, let me pay respectful homage to the late Sri Sukumar Basuji, in which memory this lecture award memorial has been instituted. I feel really extremely happy to deliver this lecture on his memory, and at the same time I also thank to the jury that they have recognized my work and bestowed on me of this award. So this award, you know, that very prestigious for everybody because it is almost 50 years old award, it is biennial. So I feel really extremely honored and then at the same time humbled for this award, because it was my dream since last 32 years to get an award of IARI, so that dream has come true for me. Coming to the today's talk, that I will be speaking zinc for food and nutritional security. You know that this is very important topic. Why I, that tempted to work on this zinc for food and nutritional security, I will take a 1 minute. In 2003-04, I applied for Norwegian government scholarship through HRD ministry, and fortunately I got that fellowship. And then when I went there, I have a interaction with the team. The team was working on organic farming, and they were facing a micronutrient problem in the organic farming. And then they assigned me to find out the rocks and minerals which are rich in the micronutrient and that can be used as a source of micronutrient in the organic production system. So I did some incubation studies, taking that, the different rocks and minerals material, city waste compost, and compared with the fertilizers in the inert material of quartz, and collected leachate weekly, and then analyzed with the AAS and then ICP. So this was the turning point for me to work on the zinc nutrition for food and nutritional security. So after completing that study, I published that article in the Plant and Soil. It gave me a more confidence to work on the micronutrient, and especially zinc. So after returning there, I discussed with my mentor, Professor Rajendra Prasad. Sir, we have done a lot of work on the neem-coated urea, and now I want to have a zinc enriched urea for this, specifically to increase the nitrogen use efficiency and increase the zinc content in the food grains. So he said, okay, fine, that we have done a lot of work on NPK, and this is the right time that you should develop some material in our laboratory and estimate or evaluate its performance under different crops and cropping systems. So this was the turning point for me to switch over from, that nitrogen to the zinc research in different crops and cropping systems. If you see this slide, zinc is really important for the Indian scenario. And if you see this zinc concentration or zinc content in the serum of the Indian population, it is a serious issue, specifically in the rural areas where the people are not having the diversity in the food. And villages specifically more affected than the urban areas. Preschool going children having a 17% deficiency, and school going children they are having a 16%, and adolescents they are having a 31% productive population affected. Globally, now we are having 8.1 billion people, and out of that 8.1 billion people, 2 billion people are suffering because of this zinc deficiency. And out of 2 billion people, 1.6 billion children, reproductive women, they are having a zinc deficiency. And annually, if you see this 8 lakh deaths are occurred because of that zinc deficiency, and out of 8 lakh, 4.5 lakh children, they die every year because of zinc deficiency. So zinc is really very important, and it is a cost effective for that to overcome the malnutrition. And United Nations, they have said that zinc saves the life. It is a life-saving commodity. I have taken this report from different sources, and low zinc also lead to the pneumonia, diarrhea, morbidity, stunted growth, malaria, dengue, low intellectual development, wasting. So these are the the repercussions of that low zinc deficiency. I would like to take a 1 minute for this slide. You know that this slide, zinc deficiency in human beings. Dr. A.S. Prasad, Anand Singh Prasad, he was from Bihar. He was posted in Egypt and Iran, and first time he reported the zinc deficiency syndromes in the humans. This is the only, that article which I could locate in the literature, that zinc deficiency first time reported by an Indian, and that too in that he was working in that World Health Organization project in Egypt, Iran and Egypt. And at that time, after that he published another article in Journal of Laboratory and Clinical Medicine in 1963. And this article reproduced in 1990 again as a landmark article in the same journal. And he reported in this article, if you see this picture, you know that almost 18 to 21 years old adolescents, but they look like that 4 or 5 years old children. And this is the original picture which I have taken from that article. And this is the photographs of the Anand Singh Prasad, and these are the typical deficiency symptoms in the human population of the zinc. Coming to the other aspect of the zinc, zinc is not only involved in the human nutrition, but also it is having a lot of other multifaceted uses like green building, renewable energy, infrastructure, life-saving, transport, energy storage, manufacturing efficiency, and ultimately we need a nutritional security for the zinc. And zinc market globally is more than around 40 billion in all aspects. But generally, that if you see this other sectors, or we can say the service sector or manufacturing sectors is more, only 2% is the miscellaneous use of the zinc, including zinc in the human or including in the fertilizers. So coming to the zinc deficiency in soils in human beings, it is interlinked. If we are having a deficiency in soil, then definitely we can have the expectations of the zinc deficiency in the human beings. And if you see this, our country, it is almost inadequate zinc intake, about 25%, and stunted growth is 20%. And this again has been reported in other figures. Then coming to the zinc deficiency, as per the report of Wessels and his associates, 2014, again our country showed that we are having a zinc deficiency 30 to 40%. So it is a serious issue for us. It is not so simple solution. This is the latest map developed by the Shukla and his associates from the Indian Institute of Soil Science, Bhopal. Now he is the Vice Chancellor. So he stated that 63% zinc deficiency in specifically, that Tamil Nadu, and then we are having in Madhya Pradesh 57% and 56% in Rajasthan. So this is the latest data in our Indian scenario. So cereals and human consumption, they are interlinked. And in our situation, we are having the more cereal consumption, and cereals are the major source of rural energy or rural people intake. Rice, wheat, mostly important food chain in our food distribution system, and 80 crore people, they are getting a free ration now, and up to that 5 years we have extended our government free public distribution system of rice, wheat. But generally the rice content in the zinc content in the rice, in the polished grain, it is 8 to 14 milligram per kg, and in wheat it is 15 to 45 milligram per kg. And in maize also zinc content even less than the wheat. And although maize is not the staple food, but at the same time it comes into the food chain specifically for the feed, animal feed. And whatever we are eating from the animals, that comes under, that again our food chain. Storage is also equally important. Now we are 140 crore people. At the same time, we are also having the largest bovine in our country. 536 million cattle or bovine population is there. So every 2 human beings have a 1 cattle. So storage is also equally important for us. So for better nutrition for human beings, cereal grains should contain around 40 to 60 milligram. But the current situation is 10 to 30 milligram per kg. So Again, you know that rice, wheat, these are the important, and 75% daily calorie intake comes from the rice and wheat in our rural areas. Then hidden hunger. Many people, they are suffering from the zinc deficiency, including me. I am also taking a tablet of zinc because I have tested blood serum. So many people may not be knowing they are having zinc deficiency. So hidden hunger is one of the important components for the micronutrient deficiency. So we have increased the high productivity under cereal crops that will lead to the hidden hunger. We have reduced the area and then production under high protein and high micronutrient, specifically pulses, and cereal price is not as high as pulses and other, those contain the high micronutrient. So this is the scenario with the micronutrient. So what are the different strategies? There is no silver bullet which can solve our problem. We have to go by different ways to solve this micronutrient problem or hidden hunger. So one important component, I can say, dietary diversification. In cities, many people can afford that diversity in the diet, but in rural area, people cannot afford. Supplementation is very important. Now, people are taking tablets. For example, this tablet luxury, if you see that there are many fatty acids, selenium is there, calcium is there, and then wheat, green, germ oil is there, vitamin A is there, and then zinc is there. This tablet costs around 60 rupees. This tablet costs 60. Supplementation is not so easy. Then fortification, physical mixing of any micronutrient. Our government of India 2022, they have taken a decision for iron fortification in rice under public distribution system, and they have taken the 15 districts on the pilot basis. So government is taking different initiatives, but as an agriculture scientist, we can go for biofortification. So biofortification is a sustainable way where we can increase the density of any micronutrient, protein, or any other antioxidant. Among these, agronomic biofortification include the fertilizer application, and fertilizer application may be soil application, foliar application, and then we can have bioinoculants, we can have crop residue, intercropping, mixed cropping, so all these comes. So biofortification, if we describe it, it is a process of increasing the density of vitamins, minerals in a crop for plant breeding, or agronomic aspects or practices so that the biofortified crops, when consumed regularly, will generate a measurable improvement in the vitamins or minerals in the nutritional status of the human beings. So far, ICAR has developed 87 biofortified varieties in 16 crops, and some of the varieties are very promising, and some of the varieties people have taken to the farmers' field. This article published in 2014, where we have stated that agronomic biofortification and breeding approach, they should go hand in hand, because we should not work in isolation, we should work together, then only we can achieve. But biotechnological approaches may not be acceptable or may be acceptable. Just for example, golden rice, who are the takers for the golden rice? So these are the situations. So coming to my actual talk, that where we have done some of the work and generated a lot of information on agronomic biofortification. So adequate fertilization, method of fertilization, time of application, FYM, integrated nutrient management, crop residue, intercropping. These are the certain, you know, that avenues where we can work as an agronomist to increase the micronutrient efficiency. And 4 are specifically nutrient stewardship, right source, right rate, right time, right method. So these are the important considerations for the zinc biofortification agronomically. And we have tested many products of the zinc, specifically zinc sulfate, zinc oxide, slate zinc, green manure, green or zincated urea we have developed. And crop rate of application may be crop based or cropping system based, depending on the requirement. And then time of application, seed priming, or at the time of transplanting, or grain at the time of grain filling, and method soil foliar. So all these we have tried in our. You see this slide. This slides indicate the different materials which we have used. But this material, you know, that most common zinc sulfate, heptahydrate, and it has some problems because of hydration, and then it makes the lump, and then it is not free. You know that freely applicable to the farmer's field. So what we have done conditioning of the zinc sulfate of the zinc oxide and neem oil. This product we have kept up to 1 year, and we found it is very friable product and can be used effectively. And then we have also developed 0.5% zincated urea, 1% zincated urea, and we have used for our experimentation. So coming to the first experiment and some first study which we conducted taking the 0.5% and 1% zinc coated urea through zinc oxide and zinc sulfate and zinc oxide, and we found there was a significant response in terms of the grain zinc concentration and also grain yield. Here I would like to mention, initially we have estimated the zinc concentration in the whole grain including hull, white rice, kernel and bran. So we were at fault. Later on we have that overcome this problem, but initially we were not knowing, so we have done and we have shown it here. This paper was published in Nutrient Cycling Ecosystem. In 2008 we have published 3 articles on this aspect in Nutrient Cycling Ecosystem. It is a highly reputed journal. In 1 year, publishing 3 articles on same aspect. It was my journey to start on agronomic biocertification. Then coming to the second study, then based on the lead on taking 0.5% to 1% zincated urea or zinc-enriched urea, then we have developed 2%, 2.5% up to 3% in the system mode, rice wheat system mode, and we found again a very good response to the zinc concentration increase in the grain as well as stand recovery efficiency and grain yield. And this paper again published in Nutrient Cycling Ecosystem. We have also evaluated the residual effect of the zinc-enriched urea in the wheat and the rice wheat cropping system. And here I would like to make a kind attention. Zinc concentration is almost every time we have estimated it is more in the grain of wheat, but it is less in the case of rice. It is reverse in rice, and we found a very good response even in the succeeding wheat. Again, then we have done another experiment only in wheat, taking a low concentration of zinc-enriched urea, 0.5% through zinc oxide and zinc sulfate. Again, we got a very good response to this zinc-enriched urea, and almost you can see the zinc content almost 24% increase as compared to the filled urea, and also recovery efficiency more than 11%. In general, when we are going soil application, it is hardly more than 2%. It is always less than 2%. Any source you apply as a soil application. So this is again a very good, that contribution to our zinc enrichment. And this article, that we have published in European Journal of Agronomy. This is my student was Dr. Vijay Punia, who is now my colleague as a senior scientist. We took the different sources. Different sources means we took zinc-enriched urea and soil application through zinc sulfate and zinc oxide, then zinc challi and foliar application. We got a very good response to the grain yield, zinc concentration as well as extra zinc concentration. And another experiment was done again for my PhD student Dr. Shankar Jat, where we have taken a different zinc-enriched urea and at the same time we have taken a soil application. And we found that in Pusa hybrid rice 10, there was almost, that 6 milligram zinc concentration was increased because of that zinc-enriched urea. So based on this zinc-enriched urea, then we have switched over to different soil application and foliar application, taking 2 sources, zinc sulfate and EDTA zinc for enrichment of the rice grain and straw, and that paper was published in Proceedings of National Academy of Sciences, Allahabad, and we found that specifically EDTA at the rate of 0.5% foliar application at the tilling, booting stage and grain filling stage recorded almost 28% more than 28% increase in the zinc concentration, and extra 19% zinc concentration increase was recorded. Then recently we have done, that SAM scanning of electron microscope different sources of our zinc fertilizers. Like here we have taken bulk zinc oxide, here SAM of the nano zinc oxide, SAM of the urea, SAM of the 1% bulk zinc zinc and its urea, and then same of the 0.1% nano zinc oxide urea, and same of the nano zinc oxide 0.2%. And then we have evaluated its performance in the rice wheat cropping system, and we found a very good response to the different fertilizers. Specifically, grain yield increased because of this zinc oxide 1% bulk coated urea. Highest yield was recorded, and almost that 23% increase in the zinc concentration, and it is zinc concentration in the white rice kernel. It is not in whole grain, because later on we rectified our that mistake which we committed earlier. So again, it is a very good finding, and this paper was published Frontiers in Plant Science very recently, and the same response was recorded in terms of the wheat with the same product in the rice wheat cropping system. And then coming to the rate of application, this is also very equally important agronomic approach for biofortification. So we have taken some of the experiment in the other crops also, like chickpea, and we found that with the increasing of the zinc level from 0 to 7.5 kilogram per hectare, almost 30% increase in the zinc concentration was there in the grain, and 39 or 40% concentration increase in the straw. So both are equally important for us, considering the food chain in the that these products. And then we have another approach we have tested, like integrated use of summer green manure, then dual purpose summer green manure and bioinoculants in association with the microbiology division for zinc and or zinc nutrition. So coming to this first results which we have published in the Pedosphere, which is a highly reputed journal, and we have taken the different green manuring crop like Sesbania aculeata, Crotalaria gentiana, Vigna aculeata, and then we have taken the three sources: zinc sulfate heptahydrate 21%, zinc sulfate monohydrate 33%, EDTA chelated zinc 12%. And we found a very good response to the different green manuring taken as in the window period, and there was almost 13.9% grain yield increase and almost 33% that zinc concentration increase in the grain and 12% in the straw. And with the chelated zinc we got a very good response as compared to the other sources. It is obvious that EDTA is having a more efficacy for enrichment as well as increasing the productivity. Then we have also seen the residual effect in the succeeding durum wheat crop under basmati rice durum wheat cropping system, and almost same results were observed, and we have published in Biological Agriculture and Horticulture. And another, instead of taking the green manure only, we can take a dual purpose green manure, and the dual purpose green manure can be used for specifically that for pods and for grain, and their residue incorporation can increase the zinc concentration in the grain as well as straw in hybrid rice, which we have seen in our studies. Then coming to this is the landmark article published with the European Journal of Biology in association with Microbiology Division. If you see this biofortification of wheat through inoculation of plant growth promoting rhizobacteria and cyanobacteria. So what we have done, we have taken recommended dose of nitrogen and half dose of nitrogen in association of the Providencia and the cyanobacterial strains, and we found that zinc concentration was increased almost 12% because of this treatment, and also protein content 4.5%. And that can you see this picture? Can you recognize? So at that time I was very young and enthusiastic, even I was working in the field at that time, and now I have become old. So no sir, I am not young now. So another green manuring and then mediated biofortification for basmati rice. So if you see this grain yield, because of that Sesbania and cowpea, Sesbania resulted in better, and also zinc in the milled rice, almost 24% because of only Sesbania green manuring. Because that if you add green manuring, lot of microbial activities increase and they solubilize insoluble zinc to uptake by the crops. Then coming to the, that publication it has published in Frontiers in Plant Sciences. Then we have also estimated how much zinc can be added through green manuring, whether it is Susbania or Crotalaria gentiana. We found that 85 to 90 gram zinc can be added through this green manuring, and then 18% grain yield of rice can be increased and 12% wheat. And we can also increase the density of the zinc in grain of rice as well as wheat. Then this is the same representation in the graphical form, where we found that response to this Susbania green manuring. Then coming to the method and time of application, this is also equally important, and globally we are having a lot of information on this. So what we have done in the chickpea, we have taken a different, two sources, zinc sulfate and EDTA, and we have gone for soil application and foliar application, vegetative plus flowering and grain filling. So we found that, that best results with the EDTA 3 foliar spray. 53% increase in the zinc concentration in chickpea grain, and 90% increase in the concentration of zinc in the chickpea straw. So that means, and that paper was published in Communication in Soil Science Plant Analysis. And then we have also estimated the agronomic efficiency and crop recovery efficiency, because this is also equally important. And we found again that specifically EDTA 2 foliar sprays, vegetative plus foliar application, it has recorded significantly higher agronomic efficiency as well as crop recovery efficiency as compared to the other methods and time of application. We have also gone for quality improvement of oats. Oats is, that very important cereal these days. It is in highly demand in the supermarket, departmental store, specifically those who are suffering from cholesterol. So Dr. Prasad asked me to conduct one experiment on this, and we got a very good response, but we got a response in the seed coating, not the soil application before sowing or after first irrigation or before first irrigation. But when we go for 2 kg zinc, that coating onto 100 kg seed, it increased almost 44 milligram zinc, 44% increase in the zinc, agronomic efficiency, and also net return crop recovery efficiency, grain yield. And then we have also published this paper in Agriculture Research, our NAS journal. And then we have also gone for quality improvement through zinc application in maize, corn. And what we did a soil application, foliar application, a combination of both, and zincated urea. And what we observed that soil application plus foliar application of 1 kg foliar application zinc had the potential to increase the zinc concentration in the maize, corn and stover. However, recovery efficiency and agronomic efficiency was higher with the foliar application. It is obvious that when so first time we have also introduced one terminology, zinc-induced nitrogen recovery efficiency, and this was published in the Indian Journal of Fertilizers, where we have shown that, that if you are using zinc in your recommended dose of NPK, then you can increase the nitrogen use efficiency. If you see this data, so that soil application, foliar application, from 18% to 27% zinc-induced nitrogen recovery efficiency was recorded in our experimentation. We have also done some exercise to improve that this zinc sulfate heptahydrate, and we have also estimated in our field experimentation, and we found a very good response, specifically when we are going soil plus foliar 2 foliar application in terms of increasing the zinc concentration in straw as well as grain in rice, and published in Journal of Plant Nutrition. We have also recorded the crop recovery efficiency and agronomic efficiency. Again, soil and foliar application was the best option to increase the zinc concentration, recovery efficiency. Then integrated of crop establishment and nutrient management options. So in this, what we have done, we have taken the 2 crop establishment method, puddle transplanted rice, aerobic rice, and we have added 2 things in the recommended dose and reduced the recommended dose, added zinc and that microbial consortia. And we got a very good response to the, specifically when we added the zinc and microbial consortia to increase the zinc concentration in the milled rice, brown rice and hull. So This paper was published in Journal of Nutrition. Again, another study we have done in the pot experiment, where we have taken two varieties, Pusa Basmati 1121 and Pusa Hybrid Rice 10. We found a difference in the zinc concentration in both the varieties, and also NPK along with the zinc. Again, there was a significant increase in the zinc concentration. So we can conclude that there is a significant variation because of crop establishment method and nutrient interaction. Some of the work we have done, genotypic differences in the zinc response, and first we did what we did is that chickpea, we took the Kabli type variety and Desi type variety. So that means there was lot of variation. Desi type varieties, they were having a more zinc concentration as compared to the Kabli type. And again, we have taken some village variety from this institute, which developed by our honorable director, sir, like Pusa Basmati 1, Pusa Sugan 2, Pusa Sugan 3, Pusa Sugan 4, Pusa Sugan 5, and we found a very good response in terms of agronomic efficiency and zinc concentration in rough rice. We have also estimated the zinc concentration in different parts of the rice, like white rice, kandal, bran, and hull in different varieties, and we find a very that good variation in the different varieties, accumulation of the zinc. This is the difference between the productivity and different varieties as influenced by the varieties as well as the zinc application. So yield improvement was 2 to 26% in respect of the variety, and yield improvement 6.8 to 13.4% because of the zinc application. So this paper was published in the Crop Journal, again very highly reputed journal. Zinc concentration and uptake we have also estimated. So we can say that there was lot of variation in the accumulation of the zinc, and specifically I would like to emphasize that white rice kernel varied because 13 to 23% increase because of zinc application. Coming to the productivity of wheat, again we have taken different varieties, release varieties, and we found a 3.7 to 17% yield improvement because of varieties, and because of zinc application 9% to 12% increase in the zinc concentration. Then zinc uptake and improvement in the same varieties because of zinc application, and then different parts of the grain like grain or rista, and even we have estimated spike straw. So then coming to the publication, it's published in Archive of Agronomy and Soil Science. Then coming to the that biofortified, non-biofortified varieties. Recently we have estimated, and that published in Journal of Soil Science and Plant Nutrition, where we have shown that non-biofortified varieties have responded better than the biofortified varieties, BRR non-fortified. Even our that non-biofortified variety Basmati rice responded better, Basmati 1509 as compared to the BRR non-fortified. Whatever they claim 22.4 milligram, we got only 14 milligram in that variety. Sir, we have published it, and we have documented it in our ecology. Then this is the yield, that response because of fertilizer application and variety. Then we have seen also that zinc concentration in white rice kernel, and then because of varietal variation and zinc application, again our Pusa Basmati, this Pusa Basmati 1509 is performed better, and zinc application, soil plus foliar application. Then this article was published in Journal of Soil Science and Plant Nutrition. Then we have also seen that biofortified, non-biofortified wheat varieties. Here that biofortified varieties even did well as compared to the rice varieties, and soil plus foliar application was the best option. For we have seen the phytic acid and relationship between phytic acid and that zinc application. So there was a negative correlation between the phytic acid and zinc concentration, and recently this article has published in Journal of Food Composition and Analysis. And then we have also, this is the last slide, where that we have seen that even sulfur application can enhance the zinc content in the rice and wheat cropping system. And coming to the benefits of the resulting from the use of zinc enriched seed, I can say that there is a that better seed and vigor viability, improvement in the abiotic stress, higher zinc yield under zinc deficiency, improving in the resistance and seedling, decrease in the seedling rate, and ultimately leading to the human nutrition. So coming to the international recognition for our research work, in 2024 we have published in Advances in Agronomy one article. One article we have published in Encyclopedia of Soil Science. It is a great honor for us that we have published in Encyclopedia of Soil Science. And on 29th January this year, our another article on the same aspect, biofortification, has been accepted in Advances in Agronomy. So I can say that it is the most satisfying thing for me. And impact of R&D on that of our research. We have encouraged the young generation to work specifically micronutrient and in general zinc nutrition related. I have guided 8 PhD students on zinc nutrition only in rice wheat cropping system. I have motivated other scientists by delivering lectures, sharing information, and work on zinc has been widely referred by around the globe. What we have done as an impact, if you see, this is our urea. From urea to we have gone to the neem oil coated urea and then sulfur coated urea. And sulfur coated urea, you know that on 9th February 2023, sir was present there at that time when Ministry of Chemicals and Fertilizer, they made one presentation. I also made a presentation, and they said that it is your work, we want to take to the stakeholders, farmers, and now this sulfur coated urea is in the market. So this is impact of our research, but I have not highlighted for this work. And another, what we need a zinc coated urea. It should come in the market for the benefit of the farmers to increase the productivity and nutritional quality. This is my citation till date. More than 9,000 has been. So I feel happy. So future line of research, considering this research benefit, zincated urea needs to be extended from rice wheat to another, and we must develop the zinc efficient varieties. And this is my suggestion. This is my suggestion that we should work together, plant breeders, soil scientists, physiologists, agronomists, in tandem to tackle this micronutrient zinc deficiency from the food chain. And this is the time to acknowledge. First of all, I would like to acknowledge to the former directors and present director, sir, former professor and head division of agronomy, Dr. Rajendra Prasad, who is my mentor till date, and then Dinesh Kumar, technical staff, Madan Pal and Pyare Singh. Pyare Singh, he is a lab assistant. He is not a lab assistant. He is a simple, labor, fourth group, but he is operating all my analysis. He got a best worker award from this institute. I must appreciate him and all my PhD 8 students who have directly, indirectly involved. And also I would like to acknowledge our, motivator, our good colleague and then friend, Dr. Radha Prasanna, who is always with me and mentoring us. So thank you very much for your kind patience. Thank you very much.
Thank you very much, sir, for a very enlightening award lecture. Hearty congratulations once again for well-deserved Sukumar Basu Memorial Award. May I now humbly request to the esteemed chairperson for his expert remarks.
Thank you very much. Ladies and gentlemen, once again, please give a big applause, clap for Dr. Shive for delivering very, very wonderful lecture. Many of you must be thinking that what is there in zinc nutrition? For last almost 4 decades now, we are speaking of zinc nutrition. But understand this was a journey of more than 3.5 decades. He tried to just put forth you and the contributions he has made. The zinc deficiency, when Green Revolution started, after that, after couple of decades, people started off multiple nutrient deficiencies, and slowly, Dabi Awaj mein we started speaking about the micronutrient deficiency, and zinc was the first metallic nutrient which was considered to be deficit in this country. So today, although he concentrated his talk in and around zinc only, but his expertise in other nutrients also is very well known to the peer group. He has chosen the topic very close to food and nutrition security. When he started his career, that time probably food security was the concern, but subsequently nutritional security emerged as one of the important aspects of Indian agriculture and also the global agriculture. So in Indian population, as he explained, zinc is very important nutrient, and deficiency versus physiological disorders, you know very well. Now, human population or animal population, both the things, very thin line to distinguish, but deficiency in plants, deficiency maybe soils may be deficit, but the ultimate result, it brings a lot of deformities or physiological disorders or the poor functions of animal and the human being. And therefore, there is no doubt this is very important topic. Either you go plant breeding way or agronomic biofortification way, ultimate aim is to enrich the food material with sufficient amount of zinc or any metallic material that way, so that you do not suffer, the health doesn't suffer much. And for zinc fortification, not only fortification, but he also worked in coating. So he was talking of zinc coated urea now, started with neem coated urea, sulfur coated urea, zinc coated urea. Now coating itself is one of the aspects, but it doesn't provide a wider kind of solution to the complex problem. When we started speaking of food production in this country, we wanted to enhance production in 70s. That time we were not bothered about all these things. But subsequently, as our food grain production went on increasing, population rate of population, means growth rate, that also had a definite pattern in this country. Therefore, we slowly started thinking about it. And Dr. Shive very clearly brought forward the kind of school of thought he has developed, right from Dr. Rajendra Prasad, right to today, when some student must be working in this line. So in this process, several steps he has gone through, and in the process, he has developed technologies. Of course, as a result, he was awarded several times, very good number of publications, very high quality publications he has published in various journals. So all through, whether it was application part, whether it was production and productivity part, every part he has contributed a lot, and therefore I believe this award is he was the most deserving recipient for this award. So on behalf of all of you who are sitting here, I congratulate Dr. Shive for this outstanding award and for delivering an outstanding talk today. Thank you so much, and once again I'm thankful to Director IRI for inviting me and giving me opportunity to be chair of this important lecture. Thank you, each one of you.
Thank you very much, sir, for being with us and providing valuable remarks to the already enlightened lecture. As a mark of love and profound bond which we share with the esteemed chair, may I now request our Honorable Director, Sir, and Dean Ma'am to kindly do the honor of felicitating the chairperson with a shawl and memento.
Hello. Hello.
Before we reach to the end of this session, may I request our Dean Ma'am for the vote of thanks. Thank you.
Thank you, Dr. Roff. It's a privilege to accord the vote of thanks on behalf of the entire IRI family for this session. Distinguished Chair of today's session, Dr. S.K. Chaudhary, sir, Dr. A.K. Singh, Director IRI, Dr. Shive, the Professor Division of Agronomy and the proud recipient of this prestigious award, all distinguished guests in the front row, my colleague Dr. Brahmanand, Dr. Markande, our associate deans, project director, the HODs, professors, faculty, students, dear friends. It's a real, honor to be here for proposing the word of thanks. At the outset, I'm grateful to Dr. S.K. Chaudhary ji, who so promptly agreed to the request on behalf of director that we made to him, and despite his busy schedule, he agreed to be here on this session and chair this event. I'm thankful to Speaker of the Day, Dr. Shive, for delivering the wonderful memorial lecture, award lecture, which I'm sure everyone must have enjoyed in view of the practical utility that his fundamental research, fundamental and applied both together, that carries towards its implications on rice crop per se and then the nutritional aspects for the humans. So thank you very much, Dr. for your contribution. And I'm also thankful to all the distinguished guests, particularly our respected Dr. A.K. Singh sir, Dr. Jalali sir, to agree to our request of coming over here and be a part of this wonderful evening. I am particularly thankful to our source of inspiration and the mentor, Dr. A.K. Singh sir, Director IRI, whose support and guidance in a very patient manner in every minute detail that this event actually during the planning part of this event, and for that and for everything for this Convocation Week program, my heartfelt thanks to Director IRI. My thanks are also to my JD colleagues, PDWTC, Dr. Brahmanand, HODs, professors, all faculty and students of IRI, because it is a 24/7 kind of involvement of each and every one. So it's not a one-man task, it's not a one-team task, it's a task of the entire IRI family. So I bow myself in front of all of you for your contribution that you are making in making this event, particular today's session and this entire event, as a very successful and a very memorable one. Thank you one and all. Thanks a lot.
Thank you all. This way we reach to the end of this session. We'll meet again tomorrow with the LPS lecture. Thank you.
हाँ सर, चाय का व्यवस्था, चाय नहीं थी सर।